Okay, everyone, let me welcome you all to what is now the 17th Annual Plant Biology Initiative Symposium. As I mentioned last night, this is an annual symposium where members of the sort of plant biology community here at Harvard host a symposium that focuses on a particular topic each year, and it's a rotating topic depending on the faculty that organize. So this year we are digging into, that was a bad pun and unintended, root microbe interactions, particularly in a changing world. We've got a really, really great lineup of folks, and I think it's going to be a wonderful day of talks. So first of all, the schedule for all of the speakers is on the back of your name tag. Wendy did a great job of organizing that. So if you need to know the schedule, just take a peek at your name tag. And that brings me to a really, really important thing that I want to talk about first off, which is thanking the folks that actually get this thing done, which certainly is not me. Wendy Haywood, who helps you register out front, does the lion's share of the work for this. She is phenomenal, and we really, really appreciate everything that she has done to pull this off. Yes. Hopefully she can hear out there. Also, the Arboretum staff here does a great job of getting everything set up. We really, really appreciate them, and the caterers as well. I do have a couple of quick announcements that are more logistical. So first of all, restrooms, you're going to be here all day. There are gender-specific restrooms on this floor. The men's restroom is sort of around the stairs to the right here, and the women's restroom is past the posters. You will hit doors that lock, so you can't go any farther, and that's just because we've got a lot of people in the building today. If there are also all-gender restrooms upstairs, the closest one is at the very top of the stairs. Just hook around to the right. I think that at different times of the day, we may overflow the capacity of the room, so there is also overflow seating. We've got things live streaming on televisions in the Walnut Room, which is sort of in the middle of this space, almost directly above us upstairs, that you can feel free to go in there and sort of relax and watch the talks and then come back down and interact with the speakers and others during the breaks and that sort of thing, if you would like to. And I want to highlight the poster session at lunch. So we've got a really, really great lineup of technicians and graduate students and postdocs that have a bunch of plant biology related posters. They really span the gamut of plant biology. We go from root exudates to hornwort gametophytes and everything in between. So please, I know the folks that have done these posters have worked really, really hard on this. Please make the effort at lunch to go talk to people about their research and be supportive of these really, really great early career scientists. Also, it is a wonderful day. I think the weather gods are excited about below ground ecology as well. So please feel free to use all of the outdoor space, especially during lunch and breaks. If you want to peruse outside in the courtyard or around the building, please do. I don't feel like I have to say this to a plant biology group, but the horticulture staff does a really good job maintaining the ground, so please be respectful of the plants. Many of them are rare and important. I think that's all of the logistical announcements that I have. Actually, at lunch, though, the Arboretum, if you feel intrepid, heading out the parking lot here down a gravel road gets you to the main part of the Arboretum. The crab apples are not that far away on Peters Hill if you want to use your phone app to navigate there, and they are not at peak but very beautiful. So please enjoy the Arboretum grounds. At the end of the day, the lilacs are now pretty much in full bloom or getting there, and Lilac Sunday is going to be crazy, so you can get a sneak preview on Lilac Tuesday. Okay, so I'm not going to spend a lot of time fully introducing things, but I did just want to provide a teaser as a sort of a warm-up to these talks. Below ground systems are absolutely the coolest part of the plant world, right? I think we can all agree on that here. No, I am completely biased, but I think the root system of a plant is phenomenally beautiful, right? I think it is underappreciated, but one of the most beautiful parts of the plant organism. and often makes up the vast majority of the biomass of the plant, right? Many of these, many organisms, the vast majority of what's going on in the plant is actually happening below ground that we really can't see very well, so it's really hard to appreciate, right? And these roots, of course, have lots of associated microbes. 1 gram of soil can contain billions of bacterial cells. 1 tree can associate with hundreds of mycorrhizal species, right? So there is an immense amount of diversity in the soil that help plants function, right? And so it's a beautiful ecosystem. It is a highly diverse ecosystem, and it's also an incredibly important ecosystem. There are a bunch of ways that you can sort of think about how important these root microbial interactions are, but one that is, I think we are just starting to get data accumulated on this. Recently, there have been syntheses of sort of how much of global productivity goes into these root microbial syntheses. A paper by Hawkins last year suggests that something like 13% of NPP goes to ectomycorrhizal fungi in ECM-dominated systems, about half of that into AM-dominated systems. We have been doing some work on scaling up root exudation, and mostly driven by grasslands, but there is a substantial input of carbon into the soil that goes through root exudates, and some work that's still in progress suggests it's about 3% of NPP for symbiotic nitrogen fixation, right? So plants are spending a lot of their photosynthesized carbon on maintaining these root microbial interactions, which I think really underscores just how critically important these are. And the last thing I will say is that when you think about all of these different root microbial interactions, a really important thing to keep in mind is that they are fundamentally based on some resource exchange, right? Where the microbes are providing soil resources to the plant, and in exchange, the plant is providing photosynthesized sugar to the microbes. And the dynamics of this resource exchange can change dramatically as the environmental conditions change, right? So as human activity is perturbing the global environment through increasing CO2 and drought and warming temperatures and land use change and fertilization, it can dramatically change the sort of dynamics of these plant microbial interactions. And I think that's something that we are really just starting to appreciate and starting to dig into in sort of at the ecosystem level in our modeling work and in ecology broadly. So that is why I wanted to bring all of these speakers here together today to really start to learn about these plant microbial interactions and how they are going to impact ecosystems as ecosystems continue to change due to human activity. So with that, this year we're doing something a little different. I have strong-armed several folks in the department to do introductions for each of the speakers. So the first person I'm going to invite up is a postdoc in OAB, Ceci Prada, to introduce our first speaker.
Hi, everyone. I'm very excited to introduce the first speaker of the day, Dr. Peter Kennedy. Dr. Kennedy is a professor of plant and microbial biology at the University of Minnesota. He received his PhD from Berkeley and then continued there for a postdoc. He then started his faculty career as an assistant professor at Lewis and Clark College, before transitioning to Minnesota, there where he is now the Associate Dean for Research in Biological Science and the World Chair of Microbial Ecology. Peter is a fellow of the Mycological Society of America. He has been a Fulbright fellow twice, and his publications have received over 13,000 citations. Peter's work investigates the diversity and function of both fungal and microbial communities, with a particular emphasis on those that form symbiosis with plants. Plants Lab focused on the structure of mycorrhizal communities and the role that ectomycorrhizae and saprotrophic fungi play on the speed on on the speed of soil organic matter decomposition. So please help me welcome Dr. Kennedy, who will tell us about how shifting resource availability affect the structure and function of ectomycorrhizae symbiosis.
Thank you, Cece. Thank you so much for that kind introduction. That was great. Thank you, Ben, for organizing this awesome event. Thank you to the Arnold Arboretum for hosting this. It's really a pleasure and honor to be here. I'm quite excited and humbled by the lineup of speakers. And so I think I'm going to go ahead and just jump right in. But today I'm going to sort of talk about some stuff that my lab has done a lot of work over the years in different areas. We've done a lot of work thinking about how to identify fungi, particularly using molecular techniques, and I'm not going to talk at all about DNA. So I'm going to go in a completely different direction. I'm going to talk about mushrooms, and I'm going to talk about leaves, and I'll explain a little bit more about that. So a quick introduction and background. And then I'm going to jump into 3 studies, and I'm going to walk you through 3 studies that are, I think, related. There's a common theme, I hope, which I'll bring up. But the first is thinking about mycorrhizal types. So Ben mentioned thinking about arbuscular versus ectomycorrhizal as 2 big sort of important ways that mycorrhizae exist on the landscape and how that might mediate temperate tree nitrogen status over a century of environmental change. The second is I'll explain what this Seuss effect is for folks who don't know it, and does that help us actually track the movement of carbon into ecosystems, both above and below ground, and how that might change sort of the way that we think about how we can monitor carbon cycling. And the last I want to talk about is something that's really quite new and fresh that I'm excited about, but thinking of this idea that shifting tree resources might actually have a really large impact on the fungal community. And when I say fungal community, that's sort of writ large. And so although I promise I will focus most of my work on plant-associated microbes, there's lots of cool fungi that exist. And so I'm going to tell some saprotrophic fungal stories at the same time along with the mycorrhizal work. So I'm going to start with the take-home messages in case I don't get there at the end of the talk, just to make sure that hopefully these come through. So I think this is sort of the more larger scale, important parts of what I think is interesting about these data sets. So one in particular is that thinking about long temporal scales, I think, is really key to helping us understand the effects of global change. There are lots of sort of clever and important short-term studies, but really having many decades, even centuries worth of data can help us better understand how our ecosystems are responding than we can do if we just have these sort of 3-year or 5-year versions of understanding. I'm going to sort of wave the flag for both plant and fungal specimens in both the herbarium as well as fungarium collections and field surveys, and I think that actually the organism itself says a lot about what's happening with respect to global change, and so I encourage us all to continue to use those organisms directly if we can. And then finally, I'd like to sort of make the case that in these data sets, we've tried to put data sets together that I think together actually help us better understand ecosystem responses than if you just focus on fungi or you just focus on plants. And so the co-comparison of plant and fungal responses to global change, as well as comparisons across functional groups or guilds, particularly within fungi, I think helps us gain a better understanding of the generality of how the organisms are responding to various aspects of global change. Am I okay position-wise? Okay. So I'm going to start, before I jump into what's there, with some acknowledgments, particularly the 2 folks that I've been very, very lucky to work with. So Lauren Klein was a postdoc in my group and actually helped kick off this herbarium-based research that'll be the sort of core of what's the first and second story. And Talia Michaud, who is actually an East Coaster from Connecticut, but came out to do her PhD in Minnesota, is the person who's really the sort of the synthesizer, the deep thinker, the one who has pushed this data both into publication form, but also pushed me to think about questions in new and exciting ways. And so Talia's been fantastic. She's defending her PhD in a couple of weeks and maybe going in a different direction. So I wanted to make sure that this science got seen by folks. And so I'm excited to basically tell you a lot about Talia's thesis. I'm also going to say that the collections that have been made for both the fungi and the plants, a lot that were done in Minnesota, have been collected on the native lands of both the Dakota and the Ojibwe peoples. So with that set of acknowledgments, I think it's, as Ben said, we live in this sort of important and rapidly changing time. So the Anthropocene is one where lots of global change factors are happening. A lot of them are concurrent, a lot of them are interacting, and so you can look at many of these different graphs, and you can see, of course, here there's the sort of classic CO2 rise. That CO2 rise with the red lines is concurrent with the change in temperature relative to sort of the average temperature in the last century. This is an example of, say, the amount of nitrogen that's being deposited into ecosystems at different sort of projected or actual measures of all these factors. And so thinking about these concurrent factors, how they're affecting these ecosystems, is something that I think we are really sort of fundamentally faced with, no matter what kind of question we want to ask specifically. So I think a lot of the way that we've tried to tackle this has been through observations and experiments, which is 100% important and valid as a way to understand how our ecosystems are responding. So again, you can go out and you can monitor forests, and you can try to measure lots of different things. You can manipulate that. So that's a picture of the Duke face experiment. These are important, and I do lots of these in my lab. I will say that oftentimes there are limitations to what we can understand by doing that kind of work, right? So often this is sort of single site based, and so my forest might be different than your forest. Oftentimes we're doing them on relatively short scales in terms of time, and so inference can be limited. Not to say that it is important. So you can get around that in some ways. In meta-analyses, Colin talked about this yesterday, there are lots of important opportunities that meta-analyses give us to have, sort of a larger understanding. Again, you can incorporate multiple sites, you can look across timescales, but oftentimes you really don't get that sort of time perspective in the meta-analyses. You usually get a better sort of spatial inference from those. So I actually think that herbaria is a really underutilized resource to ask questions about global change. I'm just going to quickly say this is a great study if you want to jump in on why fungi, and there's a whole discussion of what is a fungarium. I'm not going to get into that here, but there's really a lot that can be asked and excitingly answered, and I hope that my talk sort of gives you some inspiration for that. There's a couple of things that I think herbaria can do particularly well. One is we can extend the time scale, right? So we can go back multiple decades. In fact, we can go back multiple centuries, depending on how the collections are. And we can also look at herbarium collections from lots of places to get a broader spatial sense of what things look like. And then, as I mentioned earlier, we can co-analyze data sets together to sort of see if the same patterns are showing up in plants as well as in fungi. And that itself can, I think, generate broader inference. Okay, so that's the background. That's kind of where I think this science is pointing. And so let's jump into this first study. And so thinking about mycorrhizal types and how it mediates nitrogen status in temperate forests. This is a great synthesis paper from a few years ago in Science that basically is showing that we are progressively limiting the amount of nitrogen that exists in our ecosystems. And there's lots of different ways and interpretations for that. A lot of that probably has to do with the CO2 that we're pumping into the atmosphere. But there's this pattern of oligotrophication that is consistent in lots of places. And we know that mycorrhizal type has been increasingly invoked to predict forest responses to environmental change. There's lots of great work. I'm going to feature just a couple of stories, but there are many. One from Colin, of course, is this neat study where he looked across the continental US, looked at basically AM and EM type forests, and looked at, in this case, recruitment. And you're seeing that across the nitrogen deposition gradient, there's a real shift in which of these two mycorrhizal types the plants are doing well in terms of recruitment. So increasing N deposition tends to decrease ectomycorrhizal recruitment and increase AM mycorrhizal recruitment. It seems like a lot of that has to do with sort of the capacity of the fungi to either access organic or inorganic nitrogen, as well as their sensitivity to that nitrogen. This is a neat meta-analysis that's been out. There's other, I think, versions of this, but basically suggesting that when you look at these mycorrhizal types and you look at the cross between the effects of adding carbon dioxide into your atmosphere, and if you're in a low versus a high nitrogen environment, you see these differences where it looks like ectomycorrhizal fungi can really sort of, or at least ectomycorrhizal plants can do well in both high and low nitrogen environments when CO2 is changed, versus it looks like maybe there's a different response for the AM trees and plants that were in that meta-analysis. So there's kind of conflicting ways to think about, can ectos be this sort of help to plants in a high CO2 world, or with nitrogen deposition, are we going to see lots more of these AM plants? So we asked kind of a simple set of research questions. The first was really to look at both plant nitrogen concentration and the delta 15N, so the isotopic signature, in both leaves as well as in fungal collections over a long time scale, so actually 14 decades worth of data. We were trying to understand how much mycorrhizal type, so whether it's actomycorrhizal or AM, in this case, trees in this data set, but also ectomycorrhizal fungi versus saprotrophic fungi, how much some of this trends we can track by type. And then finally thinking a little bit about sort of the interpretation. So what is it about the mycorrhizal mediation of particularly delta 15N that can help us understand the results? So this is a set of herbarium collections. There's actually about 500, about 300 plant collections, about 200 fungal collections. You can see sort of the distribution of which plant species. These are all hardwood trees of various sorts in the greater 7-county metro area of the Twin Cities, so localized to Minnesota. And so you can see the sort of distribution of the collections, whether they're ectomycorrhizal fungus or plants, and whether they're non-ectomycorrhizal, meaning saprotrophic fungi, or arbuscular mycorrhizal trees. And then here's the distribution of the fungi that are also in those two different trophic modes. So this is all happening in the background of a lot of N deposition. So Minnesota received a lot of NDEP. It looks like about a 6-fold increase over the range of that 14-decade span, as well as a major increase, of course, in CO2 concentrations, which is also happening and much more consistent globally. So in the face of increasing nitrogen deposition and CO2, what's happening to plant N status? So I previewed this and sort of talking about that just a minute ago, but there's sort of some trends that we might think in terms of interpretation. So if we look at, in this case, time, and so starting back in the 1870s and moving forward, if EM plants are able to maintain plant N through their capacity to potentially access organic nitrogen in a higher CO2 world, maybe those plants do relatively well or stay stable, versus AM plants that don't have that same access might decline. Kind of conversely, AM plants, because they can seemingly do better in sort of high inorganic nitrogen settings, maybe they actually do well against this sort of background of overall decline in N availability relative to EM plants. But there's also the possibility that this might be the trend that is what we're seeing very consistently, is that both AM and EM plants are declining in their N concentrations due to this sort of global oligotrophication pattern. And then below ground, thinking about the fungi, or in this case we're looking at mushrooms, but thinking about the mycelium, there could be trends where both saprotrophic and EM fungi, they might also be declining, right? If this is really something that's sort of ecosystem-wide. Potentially, though, fungi might be able to hold on to, so they can go in and get access to nitrogen that plants can't, and they may actually hold on to it. And if they do, maybe their sort of N concentrations or their delta 15 and signatures are more stable over time, whereas plants are dropping. So this is sort of the range of predictions when Talia and I were first thinking about this with Lauren, and this is the first of the results. And so we're looking at N concentration on the, in this case, y-axis. We're looking at time, so this should go from the 1870s. This is a multi-panel figure, so that the time scale will emerge in the lower panels here. But basically what we saw was that it's really pretty clear that the concentration of N, it declined in all groups, interestingly, except for saprotrophic fungi. Turns out that lots of the saprotrophic fungi in our data sets are wood rot fungi that I'll come back to in study 2. They might be decoupled from soil N availability. There might be sort of a different dynamic there. But for the most part, we're seeing a decline in all of these cases. If you look at the delta 15N of these different groups, again, we're seeing the same thing that we see with the N concentration data. Overall, we're seeing a decline. But interestingly, there's a slight difference here in what's going on with the AM versus the EM plants. And so there's a difference in slope for the EM plants relative to the AM plants. But again, all of these are showing decline. So that was sort of the first major conclusion. And questioning this mycorrhizal type, this result suggests maybe there is something to mycorrhizal type that's important to pay attention to. So we've dug in a little bit more to thinking about what that is and trying to think about sort of how these delta 15N signatures change. Joe Crane and many others have synthesized this data from many thousands of, in this case, plant collections looking at delta 15N. And generally, we tend to see a lower delta 15N signature in ectoplants than we see in AM plants. And what's interesting is that this is also, there's some neat work that's been done experimentally to show that the concentration, or in this case, the isotope ratio in needles changes depending on the biomass of ectomycorrhizal fungi. And so where there's less biomass of ectomycorrhizal fungi, we actually see higher delta 15N signatures. And so the way we're thinking about that is why the EM really are not sort of declining maybe at the same level is that it seems like maybe there's this effect that we're running into where lots of these nitrogen-sensitive ectomycorrhizal taxa, which seem to be more sensitive than AM fungi, are changing some of their functioning. And so we can imagine that if nitrogen deposition is really having some detrimental effect on the EM fungal communities, that might actually then lower the biomass, and with lower biomass, you'd see this change in the needle nitrogen concentration. And so you'd actually kind of have more of that sort of higher level of delta 15 signature over time, and that would explain why there's that sort of shallower slope in the first result. So we wanted to ask this question elsewhere, and so actually we just went ahead and took this larger data set that Joe had, and we subset it to temperate forests in the continental US. They're collections from lots of different places. The first part of that was just in Minnesota, but we looked elsewhere, and we could look across sort of CO2 gradients, we could look across N deposition gradients, and what we saw again was this sort of convergence where the difference between ecto and AM plants is really, they're becoming more similar over time. And so if you look across spatially a large N deposition gradient across all these different sites, you can see that the difference in delta 15N between the ectos and the AM leaves, in this case, is getting lessened over that N deposition gradient, suggesting that N dep is probably a lot of what that larger trend is that we're seeing in that time signal. So if you want to get into the weeds of this, this is already out and published, and so that's Talia's first chapter. I think I got to pick up the pace a little bit if I'm going to wrap up by 10:45. So I'm going to move through this a little bit quicker, but I think I can get to at least, hopefully, these two other studies. So no surprise, we're adding lots of, in this case, CO2 to our environment, particularly through fossil fuels. This is a great website that talks a lot about these carbon dynamics, but you can see this large-scale increase, particularly since about 1870. There's also in our atmosphere a major change in the delta 13C signature that we are experiencing. And so we can see here there's this decline. A lot of this has to do with the fact that we're pumping, in this case, CO2 into our environment, which has more 12C than 13C. And so that's changing the isotope ratio. And so we're actually ending up with a more depleted signal, or a more negative signal in this case. And that is steepened significantly in the past, say, 60 years. And so this is what's referred to as the Seuss effect. There's other ways to look at that. You can look at 14C ratios as well. But we were curious to say, what does it look like when you think about plants and fungi? And so can we actually track this into forest leaves? Can we track this into the sporocarps of different fungal functional groups? Does that break point, does that look similar or different, depending on whether you're an ectomycorrhizal fungus or a saprotrophic fungus? And then finally, if you look within saprotrophic fungi, does the substrate at which that, in this case, fungus is eating, particularly in this case, a plant, determine the delta 13C slope? So we actually used the almost exact data set that we had from the first study. So we had all this great long-term data of 13C records from the same collections, but we supplemented this, and Talia had the idea that we should look at these additional taxa. So there are some fungi that are specific in where they are growing on wood. There are some that really only grow on twigs of trees, or dead trees in particular, and there are others that will grow on the logs or the trunks of dead trees, and they're actually quite different in terms of sort of where they show up. And so those have different carbon ages in them. Twigs, of course, are much more recent versus much older for the logs, which are integrating a much longer time scale. So the predictions in this case, you could imagine that, again, over time, what's happening to the 13C signature? Well, we know this is what's going on in the atmosphere. And so we would expect for ectomycorrhizal fungi, which you think a lot of that carbon that's making their mushrooms is recent photosynthate, it should be dropping significantly over time. The same thing for plants, of course, they're pulling that CO2 right directly out of air, and so they should really track the atmosphere pretty closely. So both trees and EM fungi would look a lot more like the atmosphere versus a saprotrophic fungus, which might be using potentially some of that older carbon, is going to have a more shallow slope, right? Because they're accessing, in this case, less recently fixed photosynthate. So what did we see? So this is sort of the quick look at things. And so from the sap side, we do see, in fact, a much shallower slope than we see for both the plant as well as for the ectomycorrhizal specimens that we had. Interestingly, if you look at those slopes, the plants and the ectos don't differ, but the slopes are a little different. And part of that has to do with the fact that we think ectos are good at actually accessing organic nitrogen. That organic nitrogen has components of it that are carbon, older carbon, and so that might sort of make that slope more shallow as well. So this is the breakpoint analysis. So that's the atmosphere. And so we have this interesting breakpoint where, again, with the Seuss effect, about 1957 is when things break. And we see for plants that really holds up. Plus or minus, sort of we put some confidence intervals around that. It was right about that same time. So we see a match between the plant and the atmosphere. What about for the fungi? Unfortunately for our ectomycorrhizal data, we really kind of have a sparse data set, particularly early and around the breakpoint. And so the analysis doesn't perform very well because of the sort of smaller and disjunct nature of our data. The saprotrophs, however, do look interestingly different. So if you look at where this green line is and where this green line is, there is a significant difference. It looks like the saprotrophic fungi in this data set, which in this case are a lot of wood rotters, are accessing carbon on average that's about 44 years older, plus or minus 20 years in each direction. And so that seed came into the ecosystem many years prior relative to the seed that's coming into the ecosystem for the plants and for the ectos. So to really kind of nail down that the timing of when C entered the ecosystem matters, we can ask this question between the twig rotters and the trunk or log rotters. And so we would expect that the log rotters, again, would be using that older carbon, and so they would have in this case, more shallow slope, whereas the twig rotters are, again, using that newer carbon, which, of course, has that steeper slope. And sure enough, that's what holds up, which was exciting to see. And so it looks like the log rotters, on average, do have a decline, but it's significantly less than what we see for the twig rotters. And so that difference suggests, again, that these plants are incorporating this carbon, but then the fungi are accessing that carbon as to when it was brought into that ecosystem. So am I okay? Okay. I'll still try to work through this relatively quickly to keep us on schedule, but I do want to spotlight this because this has been a fun and sort of new way to think about, in my opinion, what's going on and how actually what's happening above and below ground might be really interestingly synchronized in ways that we haven't, I think, recognized. So I think all of you are familiar with this phenomenon it's referred to as masting, right? So this is the mass production of seeds in an asynchronous, temporally time pattern, and so you get lots and lots of production of, say, oak acorns or beech nuts, or you name your tree that does this, but there are many, many species that do this. So these large seed crops actually are put out in various intervals, and you can actually see the effects of this in terms of that massive resource input into the seeds where there's this switch by the plants. So they're not growing as much. And so you can see actually in this case the tree rings of the mast year relative to the tree rings of the years when there's not a mast, that all of those resources have now been put basically into the seeds. And so thinking about fungi and fungi that want to use carbon that comes from trees, as well as just the larger forest ecosystem, what does this resource draw mean for, in this case, the fungal community? We got excited about thinking about, could we analyze and think about ectomycorrhizal fungi, and there, in this case, the amount of mushrooms produced, or the diversity of that community. And you would think that, again, because the carbon is really directly tied to, these are functionally obligate, in this case, in their symbiosis. So they're not out getting carbon from other sources in a significant way, that as more seeds are produced, you're getting less numbers of mushrooms made because there's less carbon that's being pumped below ground. You might think in this case that the saprotrophic fungi really are sort of, less tied to direct incorporation of recent photosynthate. There's enough litter on the ground that they can sort of buffer themselves across a mast and a non-mast year. So in terms of this, we, again, sort of just have a couple of questions here. So thinking about plant seed production and then fungal spore carp production, how are those two linked together? To our knowledge, I don't think anybody's really looked at this, and part of it is because we're lucky to get access to some really unique data sets. And then how much does this vary by guild, right? Is it an ecto or a sap thing, or is there actually this larger ecosystem response? And then thinking about not just sort of how many mushrooms there are, but is there any community effects? Do we see some taxa that are really particularly affected by mast years relative to others? So this was a kind of awesomely serendipitous meeting on a totally different project where we had somebody who was involved in this mast tree database, a guy named Ian Pierce, who's a fantastic ecologist based at the USGS in Colorado. And Ian and Talia were talking, and they were talking about mushrooms and masting, and sort of spun up this, wow, is there this link? What's going on? And so we spent some time trying to figure out where could we get a good masting data set and where could we get a good mushroom-based data set that we could have for multiple years. Because masting happens very incrementally, right? So it's once every 3, 4, 6 years. And so you need these sort of long-term data sets. And so we lucked out in that there is really good records from Europe for European beech of its masting patterns. And there's also this amazing spore carp data set. So there's about 27 years of collections, 100,000 mushrooms. Simone Egli was the lead on this, but they were tracking the number of species, the number of fruit bodies in La Chênaie, which is a beech-dominated forest in Switzerland. And so we pulled this together, working with Ian, You can look at sort of what the MAT and MAP patterns are over time. And so you can see that there is an increase overall in temperature, which of course might affect fungal fruiting, right? Because warmer temperatures tend to be favorable to fungal growth. MAT looks like it's relatively, with a lot of variation, kind of not changing significantly over the time the data. And then this is an index I don't have time to get into, but basically showing that there are some years where there's lots of seeds produced, so big kind of like massed years versus years where there's not as many. And so this gives us that nice sort of x-axis of gradient, in this case, seed production. Importantly, seed production is not strongly correlated with MAT and MAP. So when we look at seed production, we feel pretty good about not suggesting that we're sort of co-varying with things like precipitation, which, wetter conditions might also favor fungal production. So this is the kind of one of the big high-level pictures that comes out. It looks like in heavy mast years, if you look at big mast years versus much lower, in this case seed production years, there's a massive decline in sporocarps, right? Half of the mushrooms in the forest just aren't there in high seed production years. Now the question is, does that vary between these two guilds, right? So we're expecting for ectos, we're going to see this steep decline, and for saps, we might not see the same thing. We actually don't see any significant difference in these slopes. In both cases, we're seeing that resource effort and allocation and draw that trees are pulling, not only is it affecting actomycorrhizal sporocarps, but it's also affecting the saprotrophic fungal community as well. You can track this into, so it seems like this is maybe an ecosystem level sort of phenomenon that's important to consider. You could track this into the diversity of the community. You can track this into the richness of the community. There are declines, right? So you are shifting not only the number of mushrooms, but the types of mushrooms that are showing, which taxa are there in abundance. And if you look at sort of explaining the composition of these fungal communities, there's lots of variation explained by things like MAP, but there's this sort of interesting inverse correlation. So, in very wet years, that's explaining some amount of variation, but in very dry years actually look a lot like high seed years. And so we actually see some similarity in the communities that are showing up in drought years relative to the communities that are also associated with high seed production years. And so there's a water story that I think is part of this, in addition to a carbon story. I think it has to do with this large draw that actually filling all those seeds up with biomass takes a lot of water as well. But getting the monitoring of these soils for both nitrogen and water status over that same time is a dream. Not there yet, but it would be really neat to kind of continue to push on that. So the takeaways from study 3 are that you often think about masting as this input in the ecosystem of this big resource pulse, and then there's lots of seed predators and other things that happen in food webs. I think our results are hopefully pointing us towards this other interesting hypothesis that actually resource availability can be thought of as the switching has this negative effect on the fungi. And it's true of soil fungi, both the saps and the ectos from what we're seeing. And so what I think is actually that rhythm that we see above ground, right, with this pulse of resources that the seeds are there, there's lots of seed predators, it might also be happening below ground at the same time in this case. So with that, I think I'll go ahead and wrap up and say thanks to lots of folks, great students. Been lucky to have some funding as well as many collaborators. And if there's any time, I'd be happy to take a question or two. Thank you. Pam.
Great talk. That's awesome.
Thanks.
I have a question about the first two parts.
Sure.
They're related. I'm curious if you looked at the relationships for the first part between N15 of the plants and the two forms of mycorrhizae with deposition on the x-axis for the years you had it, or soil head if you had it. And then similarly, have you looked at the C13 of the plants and the various mycorrhizal pools regressed against C13 of the atmosphere?
We have that data. I do actually have a slide in here that shows kind of what that trend looks like. So you can definitely see that this is local to Minnesota, and so we can model the increase in CO2. And so we have that specific data for all of the different time collections as well as for N-load. And so N-load is changing over this time period. It's stabilizing to some extent in the later times. Both of those are important and have strong effects locally in terms of explaining both delta 15N. In terms of their specific correlations with N concentrations, I don't remember them being dramatically different from some of the patterns that we were sort of seeing when we looked at sort of just the year alone. And so I'll have to look back. We might have that in some of the supplemental data. We did try to take that into some of the larger data set number 2 that was that larger sort of continental scale. We see basically kind of a similar pattern where across NDEP, the EM-AM convergence is still there, both in Minnesota and at that larger scale. So yeah, I don't remember the exact relationships that were there, but I think they were pretty consistent when we looked at them in both cases.
So in your third part, do you think that synchronicity between the EM fungi and saprotropes suggests some sort of cooperative interaction? Like you need that carbon flowing through the ectos to juice the saps up as well?
Huh. That's a cool possible interpretation. We talked about the idea of trying to throw in some sort of like trenching treatment or something that might, girdling, that would manipulate those in mast years to see how much that is sort of a coupled effect.
You might expect it to be opposite if they're
Yep.
repeating, right?
Yeah. So I think it's an interesting hypothesis. I think we've definitely kind of come at it more from the competition angle and sort of the broad resource scale limitation, where it really seems like the plant draw on nitrogen and water is just so immense that it really makes the ectos and the saps respond the same way. But it seems like it's possible, and there's certainly priming effects that have been well shown. You've shown a little bit of that as well. That suggests that ectos can add carbon below ground that really makes saps do things better than when they're not doing as well. So yeah.
Yeah, you have one slide where you talk about non-mycorrhizal species, and I'm thinking about invasive species and how are they responding to nitrogen deposition differently than the mycorrhizal species. I think that's a good slide.
Yeah, it's a great question. In this case, we really, the only thing that we had that was non-mycorrhizal was the saprotrophic fungi. So we either had arbuscular or ectomycorrhizal plants. But your question broadly is a great one, right? Is there something about the symbiosis that may change the response of invasive plants? A lot of these plants, they will find new fungal partners in these new landscapes. There are certainly, non-mycorrhizal plants that tend to do well in disturbed ecosystems. That functionally replaces the need for paying carbon to these, symbionts, and so they can do well in, I would say, more nitrogen-rich environments typically. How all three of those sort of connect together, I would think that certainly, yeah, if you can invest your carbon in growing and you have plenty of nitrogen and you don't have to pay your fungi, your capacity to invade would be higher. But I don't know of a really rich data set that would allow you to make kind of that large-scale generalization. Interesting question for sure.
All right, great. Well, let's thank Peter once again. That's great.
Turn it off. Thanks, man.
Go for it. Go for it.
Hi everybody, my name is Nikhil. I'm a graduate student here at the Arboretum. Today I have the distinct pleasure of introducing Dr. Philip Poole. Dr. Poole is a professor of plant microbiology at Oxford University, where he's also the section head of the molecular plant biology program. Dr. Poole's area of expertise is in the formation and function of the nitrogen-fixing legume rhizobia symbiosis. Through his work, Dr. Poole has been published in many of the top scientific plant science and microbiology journals, and he has been highly successful at being supported by the BBSRC, which, if you're curious, that is the United Kingdom equivalent of the National Science Foundation. And recently he led a large multi-group program funded through the BBSRC with the aim to transfer nitrogen fixation to plant colonizing bacteria. Now, of course, Dr. Poole doesn't only work on nitrogen fixation, so I've had a look at his publications, and he's also published on root exudation, including really cool work with these luminescent biosensors to measure the exudation of different compounds in the lab. So really cool, high-tech stuff that has really led efforts to substantially improve our biochemical and omics approaches to studying these plant microbial symbioses that all of us think about. So today, Dr. Poole is going to be talking about life cycle transitions of rhizobia from bacteria to nitrogen-fixing symbionts. I hope you'll join me in giving him a warm welcome here today.
Okay. I'll see if I can muck it up very quickly. Well, thanks very much for the introduction, and thanks very much for inviting me. It's a great pleasure to actually arrive after having had one plane that was considered faulty and they wouldn't let us get on, and had to wait a day at Heathrow, which is not exactly a joyful experience. So it's much nicer to be in the beautiful weather here in Boston. So I hope this is not too much of a scene change, because I'm going to deal with a very specific system. But I think there's a lot we can learn from the interaction between rhizobia and legumes in terms of what it means for plant microbe interactions generally, and the sheer detail of information that we've been able to build up, which also relates to the sort of ecosystem function as well. But it's going to be fairly molecular in what I'm going to talk about. So I'm just going to have a very quick sort of recap of something I guess most of you will have come across at some level, and it's this idea of rhizobia interacting very specifically with the roots of legumes. And this has one of the best characterized, if not the best characterized, signal exchange between plants and microbes. So the plants produce a group of flavonoids, the legumes produce flavonoids which are detected by the rhizobia, and they induce the synthesis of lipocyto-oligosaccharides, which effectively are just little bits of fungal chitin. Because really, although I won't say much about it today, the interaction between rhizobia and a legume is really that rhizobia piggybacked onto the AM fungal interaction with plants. And they're basically they're pretending they're fungi, and they're using the same signaling pathway to interact with plants. So, but the first stage after this signaling is the bacteria attached to root hairs generally. There are other infection mechanisms, but generally, and this is really important, it's normally a single organism, a single bacterium of a single strain attaching to a root hair, and the plant responds by curling around the bacteria and forming a little infection pocket. And that's really important because a hole will effectively be drilled in the side of the root hair, and an infection thread will develop, which is basically a modified cell wall down which the bacteria effectively grow. And it's really important to get a curl, because you can imagine if you drill a hole in the side of any plant cell, it basically explodes under turgor pressure. So you have to actually curl and form a sealed curl for the bacteria to be able to enter this infection thread. And at this point, the microbe is extracellular still. So this modified cell wall, this infection thread, which is an exact equivalent to what we see in AM fungi. It's the same sort of structure, this infection thread from the infection of a mycorrhizal fungi. And it grows down, this infection thread grows down towards the cortical cells in the root. What's important though, and this is something that's always interesting to try and get across to undergraduates, is that while this infection process is happening, the plant is actually forming the nodule. And the infection thread is growing down towards the growing nodule. So the two things are happening simultaneously. The plant cells in the cortex are dividing to form a nodule as the root hairs are coming down to meet it. Now eventually, as the nodule develops, the infection thread moves down towards the cortex, and eventually the bacteria are effectively taken up by endocytosis. And now the bacteria are intracellular, but they're surrounded by a plant membrane, which is derived from the plant plasma membrane. And what we have then is individual plant cells filled with what are now called bacteroids. And it's simply the nitrogen-fixing form of the free-living bacterium. So actually in this process we've gone from free-living bacteria in the soil, in competition as part of the microbiome, and they're going to end up inside nodules packed into cells. And this is where an actual EM from the lab is helpful. You can see the cell wall here. This is a single cell, And each of these Y-shaped cells is actually a Bacteroid. And it's surrounded by a plant membrane. So we have a very coordinated exchange now of nutrients between the microbe, which is going to fix nitrogen and deliver ammonia to the plant, and the plant, of course, providing photosynthate to the Bacteroid to be able to conduct nitrogen fixation. But what you might just note here, see this structure here, this is an infection thread. And these are basically free-living bacteria before they've infected the plant cell. So the nodule has this, in the case of P, which I work with, this continuous process of infection of new plant cells, because the meristem of the nodule doesn't die, the nodule keeps getting bigger and bigger until eventually it senesces. And so we have the two types of cells, these free-living cells, and then we have the main part of the cell now filled with these Bacteroids. And in fact, actually, by the time you have a mature nodule here represented in this cartoon, which in a P nodule is about 3 or 4 millimeters long and about 2 to 3 millimeters wide, from here down, the whole base of the nodule is packed with tens of thousands of these nitrogen-fixing Bacteroids. So in this talk, I'm just going to try and go through many of the steps in trying to understand that transition, which we've worked on for the last few years. And essentially, it's nice to show this in a circle like this. From our free-living bacteria, we can talk about attraction and communication. We can talk about adhesion onto roots, entry into these infection threads, growth down the infection threads, and then the mature nodule, and then we have to get release back out of the environment. And ultimately, it's important because this is about selection. At this stage, we're having tremendous selection between strains. And I guess I'll just go back and mention again that usually the entire population inside a nodule comes from a single strain. It's clonal. Not always, but most of the time it's clonal. And from a selection and evolutionary point of view, that turns out to be essential. Because if it wasn't clonal, in most cases, you could never actually evolve this process. And part of the reason for that is in P nodules, those nitrogen-fixing Bacteroids here are terminally differentiated. They cannot regrow out of the nodule. They've endoreduplicated their chromosome. They cannot grow outside, and it relies on these infection thread bacteria still being there and being viable to be released, which means from a selection point of view, these have to be siblings for this to be stable in an evolutionary sense. Because if they're not the same, we would not be able to maintain the population. And I'll show you some work we've done on that in a moment. So I'm just going to bore you with some genetics if you haven't come across this. There's a cool technique which a lot of people are using now, which is you take a population of bacteria and you mutate them. And you mutate them with a transposon, which essentially is barcoded. So a jumping gene that's barcoded. And you get a population, each of these single bacteria has a single transposon randomly inserted in the chromosome. But what you can do is, I've coloured them for different transposons here, and you put them through a selection. It might be through a soil selection, it might be on roots, it might be, in our case, through nodules. And it's simply that we would put them onto plants, we'd recover them off the surface of the plant, we'd recover them from nodules, and we can look at the output pool of the microbes. Now, if you're essential in that process, let's say the microbe shown here in red, you've mutated a gene which is essential, for example, to be able to survive on roots, then guess what? In the output pool, you're lost because you're dead. Or if it's for a nodule, the same thing. And so you lose it in this pool here. And there's a high-throughput sequencing technique that nobody here is interested in. I'm not interested in it either. I rely on postdocs and graduate students to understand all the maths and the bioinformatics. But you can work out which tags are lost. And so it allows you to say what genes are essential because they're lost in these output pools. And then it really comes down to just doing sensible biology. What questions are you asking? Which lifestyle stages can you look at? And we could classify the genes. These red ones are essential because they're completely lost. The ones, I'm sort of red-green colourblind, so I often get all the colours mixed up and audiences are looking at me thinking, what the heck is this crazy Australian talking about. These sort of yellowy ones here are, you can see they're a much reduced frequency. They're disadvantaged. Some are neutral, and very rare numbers are actually advantaged. The mutant is more successful coming back out at higher numbers. So what does this look like? It turns out you can form a matrix here. We did this experiment where we looked at, well, what happens for survival in the rhizosphere? You know, you take the population, then you recover the bacteria out of the rhizosphere. You recover them off the rhizoplane and ask what did you need to actually stick onto the plant root and survive in a complex environment. What would you have in the nodule bacteria? That's those bacteria that we could regrow from the nodules. They're the ones out of the infection threads versus the bacteroids here. And these are the sort of nitrogen-fixing cells. The problem with this experiment is because each nodule is formed from a single strain, to get sufficient numbers, we had to pick 150,000 nodules by hand. So I wasn't very popular about that. But fortunately, we had a visitor who came to Oxford for a year, and she picked 150,000 nodules, while more recently we've just done large mass picking experiments. So that's the disadvantage, but also part of the fun of doing this, because it does give you some nice data. What I should have said here, if you'd read papers prior to us doing this work, it would tell you there's about 40 or 50 genes that you need in a rhizobium to fix nitrogen. By the time we'd finished this study, we could come up there, in fact, about, I've forgotten how to add them all up now, there's too many numbers there, but there's about 600 genes you need to be a rhizobium, not 30 or 40. But the point is, most of the genes, of course, are required for competition against other microbes. They're required at steps which people in a pure biochemistry lab never see, because you take a single strain and you ask, is it a nitrogen fixation gene? Is it a NIF or a fix gene? Whereas most of them are required for things like sticking to roots, chemotaxis, competing against other bacteria. But it really opened up the whole ecology of what's happening inside in this association. And what was important is, we show this line here, this rhizosphere progressive line. It just means genes that if you knock them out, the bugs were uncompetitive in the rhizosphere, but you also lost them off the root, you lost them from nodule bacteria, and you lost them all the way through. So you could look at the genes which are required in a progressive sense, because those processes were essential. I'm not going to try and bore you with 600 odd genes at all, so I've just wanted to pick a few things which are fairly clear. And this was just following up on this work, and it turns out in rhizobium, I've just summarised the data as to say here it's green means it's neutral. If it's green, it doesn't have an effect. And rhizobium has two independent chemotaxis operons, which are responsible for enabling the bacteria to respond to chemical gradients. The first of these chemotaxis, one, is the thing which would affect the swimming and chemotaxis of the bacteria in laboratory media, in conditions which you could measure easily in the lab. And if you knock out those genes, our analysis showed they're terrible, obviously, at swimming, and they're terrible at competition for nodulation. They don't affect the other stages, but it's really the competition to get into the nodule in the first place. They're really important, which says these are important to swim onto those root hairs. And if you knock that out, they become non-competitive. Then we have the second system, which has never really been seen to have an effect, but remarkably we found it was actually important for bacteroid differentiation and the nodule maturation. And actually we think that it's responsible for enabling the bacteria to grow down that infection thread. But we'd never seen that before, because you could only really see that in an environment where you were doing this sort of global analysis. And that's an ongoing study to try and see, what part of the flagella motor that you have in bacteria is responsible for that. Okay, I want you to ignore this almost completely. It's just saying that as part of this sort of study, we then did a lot of microarray and now RNA-seq analysis of what's happening on the root surface to correlate it with the genes that are required. And one of the things which really turns up as a general thing you can see is you see huge numbers of stress genes are switched on roots. So it's a very stressful transition for bacteria to be on a root surface. The plant's producing a lot of antimicrobial compounds in many cases. They're often under redox stress. And this is the general stress response which we see in alpha proteobacteria. And these things are like 50 to 100-fold upregulated on the rhizoplane of our root systems. So another thing which turned up, and this led to a big study, which I'll just go over briefly, was we found a 3-gene operon is highly upregulated on the surface of roots. And I thought we started working on this because 3 genes cannot be difficult to work out what they're doing. And that was, I think we actually started on this, to be honest, about 10 years ago, and we're still confused as to what the 3 genes do. But they're really interesting because they're highly upregulated on P. They turned up to be important from this mutational study on the surface of roots. They're also upregulated on barley, and we've put them on other species as well. And we always see these genes being switched up, these 3 genes. And these 3 little genes here, one called LPP, which is for lipoprotein. One's a sigma factor, so it replaces it's an extra cytoplasmic sigma factor, which replaces the normal housekeeping sigma factor. So you need this to recognize a bunch of genes, pulls in the holoenzyme of RNA polymerase, and switches on a whole suite of genes. And this is the sigma factor here. And there's another thing, there's an anti-sigma factor. And the way these work is the anti-sigma factor normally sits in the cell membrane and basically holds on to the sigma factor. So the sigma factor cannot result in transcription of genes. It can't pull the holoenzyme down to switch genes on. You see an environmental signal, and the anti-sigma factor is either cleaved or it undergoes a conformational change, releases the sigma factor, and the sigma factor can switch on a bunch of genes. So basically, one's, if you like, the gatekeeper is the anti-sigma factor, holding on to the sigma factor until it sees an environmental signal, and then the sigma factor switches on genes. So this has got to be easy to try and understand what this does. And I wonder if that's going to, I'll just see if I can click on that, see if I can click on that one up here. There it is. If you watch, you might start to see, it's coming on. You can see here. What this is a time lapse over about 12 hours. And I hope, and it should keep repeating, and this is actually a pea plant in a petri dish. What we have here is the promoter of that 3 genes linked up to a Lux cassette, and we inoculate our bacteria. And we measure this over weeks. And I always wanted to put this, the picture of this on my bedroom wall, but my wife said that was it. That was divorce coming up. But the reason, I hope you'll notice something that is quite staggering about this. It's the spatial location of gene expression. And it's something that gets lost all the time. People mash up roots. They just look at global gene expression. And you can see here in the time lapse, it's just within about 6 hours, you start to see it accumulating around roots. And this is actually just high resolution, just showing you that when you do this, you actually have, you can see it's right in the root elongation zone that this set of genes is switching on. And so that says something really important must be happening here. And when we look at this, we can do all the sort of basic biology of this, and we can show here, this is actually where we look at how many bacteria are stuck onto roots, because you can use these Lux fusions in a different way. If you take a constitutive Lux fusion, not an inducible one of the gene, you can measure the number of bacteria on roots really easily, because you just look at luminescence in a luminometer. And we can just see how many bacteria via their counts are stuck on roots. And this shows if we knock the system out, please note this is a log scale, we get a massive drop in the attachment. And if we mutate the anti-sigma factor, which then releases the sigma factor, we then get a big increase in attachment. And this is just showing you the same thing here. This is the luminescent signals on roots. Basically, there's nothing in an ECF. It's gone crazy in the anti-sigma factor. It doesn't seem to be particularly affected by the lipoprotein here. So what we found was, if you just look at this picture over here to start with, we've constitutively labelled the bacteria with GFP, and then we've also made an inducible fusion again to the promoter, but now to cherry. And you can see it's really on the root surface. Here's the merge. It's on the root surface. It's not on the root hairs. It's not out into the rhizosphere. This is where this little operon is being switched on, right on the surface of the root. And when we just ground the roots up, we found actually it's a glycoprotein, which is what this is basically showing. We could show here, this is the amount of luminescence we get with the little bioreporter when we grind up roots. You can see it's got a nice signal here. But if we actually treat with a proteinase or an enzyme which degrades glycoprotein, we basically lose the signal. And so we know this is a plant glycoprotein. I won't go into all the detail. We know it's over 50 kilodaltons. We've done a partial purification and some mass spec analysis and have some potential targets now that we know it is a glycoprotein. We can actually take a pea seed lectin, and as long as you use a lot of the pea seed lectin, we can also stimulate the system. So what we're saying is we're pretty confident it's a lectin-like protein on the root surface, which the bacteria are detecting. It's not this particular P lectin really, but it's going to be a root lectin that we're trying to, we have 3 or 4 targets which we're about to express in tobacco leaves, because obviously if we can get the protein, then we can actually mutate the gene, then we can look at the function, and then we can really start to, probably only after 20 years, can we start thinking about publishing our first paper. So that's also an indication you do not want to do any graduate studies in my lab, because that's not true actually. We do things. But there's one other thing I want to point out, and this looks complicated. All we've done is sectioned the root in 5 chunks. And then we've looked at the amount of protein in micrograms from each chunk. And if you like, you'll notice here, this is the signal again for this luminescence of this operon. Here, we don't need to add I'm sort of at a wrong angle here, but it's about 2 micrograms of crude protein from that section will give us a very strong signal. And as we move along further, we need 10 micrograms of the protein and so on. But what I really want you to understand is that there's this relationship here that at the tip, we need 2 micrograms to switch the system on. But if we add 5 or 7.5 or 10 micrograms, we switch it off. And so we can get a bit of switch on here, but we need a higher amount than we did there. So actually what it's telling us incredibly is the concentration of the protein is like that, is highest at the tip, right? But actually as you get more protein, you actually switch the system off. So in other words, this operon is detecting a very, very narrow range of protein. Too little, you don't switch the system on. Too much, and you switch it off. And so that starts to get into this spatial ability to switch on the genes in a very narrow region of the root elongation zone. And at the moment, we're still trying to understand that, and trying to understand that in terms of the transcription of the operon versus the translation and so forth. But there's one thing I would point out from an ecological perspective, and that's to say, why does it matter that rhizobia have a way of detecting the root elongation zone of a root? Because that's what they're doing. And if you think about it, what's the only part of a root which isn't already colonized? And the answer is it's the newly growing root tip and just behind in the root elongation zone. So if you have a mechanism to find the root elongation zone, you have a phenomenal advantage to being able to colonize roots. And so that's why we think this is so important. And this is just a summary. I do not want to go into the detail. It's basically saying downstream, we found this operon. It seems to be controlling iron metabolism in the bacteria. And that seems to be how it's actually affecting attachment. But let's ignore that because it's far too complicated. This is just to point out, as we go through the, that needs a kick. This is just to show that as we go through this development process, we end up with these Bacteroid-like structures. And this is just a serial section through a nodule, and then you can see we've been able to do our proper three-dimensional representation of how the Bacteroids get packed. This is just serial section with an SEM, and then you stack it together to develop a three-dimensional image. And what's happening is there's this massive upregulation of a few genes and massive downregulation of genes in our Bacteroids. They're non-growing cells. They've switched off DNA replication. They've had massive changes in their developmental. So metabolism is radically altered, and they become what we call ammoniaplasts. If I'm getting close to time, tell me, because otherwise I'll be boring everyone for far too long. Again, this is just showing what a free-living, what rhizosphere metabolism looks like. Again, it's boring, but all I want you to notice, look what happens when we get to our early nodule bacteria. Most things are switched off, and when we get to Bacteroids, they're really just metabolizing organic acids, C4-dicarboxylates, which are provided by the plant, and this is what's fueling nitrogen fixation. So the Bacteroids are metabolically very constrained. They're more like organelles in what they're doing. And just to give a further example of that, this is something which came out of that analysis we did with mutational analysis. There are a bunch of genes in homospermidine synthesis which no one had really looked at previously, and yet they turned out to be essential they were lost if you mutated homospermidine in that selection process when we looked at what came out of the nodules. And so when we started putting individual homospermidine mutants on pea plants, this is a mock control which has got no nitrogen, no rhizobia, growing in vermiculite. This is what the wild type looks like, and I think you can see these two plants are really starting to get very sick by about 4 to 5 weeks. And if you actually look at the shoot dry weights at 6 weeks, this is a mock control. This is what happens if you're completely nitrogen starved. This is if you've got rhizobium there. It's beautiful, it's green, it's happy. And in the case of a homospermidine mutant, again, it's almost like a mock control. And if you look at the nodules, these are wild type nodules. They're nice and pink and large. Homospermidine are horrible and small and white and not fixing nitrogen. And what it's basically telling us is that rhizobia need to make a C7 homospermidine molecule to sustain nitrogen fixation. I haven't shown you all the data, but they start fixing nitrogen and then they just collapse. And I won't go into the mechanism, but it really shows you that sometimes you can then drill down into the important details from these global studies. Okay. Something that's almost old-fashioned now, but I might even be finishing with something like this, but this is something where in our lab we used to have this picture up on the lab door, and you had to work out which was the odd one out. And I think most people could work out it was the wild type, but it was this interesting experiment we did many years ago where we started knocking out amino acid transport systems in rhizobia. And we started out thinking that this thing we discovered, which is the amino acid permease, it's a very broad range amino acid transport system, transports most L amino acids, and we thought it might be important for nitrogen fixation. We mutated it, and it had absolutely no effect. And then we found there was a second transport system, a branch chain, so an amino acid transport system. We knocked that out, and we found that it had absolutely no effect. And at that point, we lost interest because it just wasn't going anywhere. Except we had, for reasons of wanting to understand the stereospecificity, generated a double mutant, an AAP-BRA mutant, which we put in the freezer. We did lots of transport studies on it and published some papers, specific papers. And then we realized one day that we'd never put it on plants. And so we took it, I got a student to take it out, put it on plants, and you could have knocked me down with a feather when they came in and showed me that they were completely nitrogen starved. So now this is sort of sat in the, as I said, in the freezer for a long time. And eventually we came in, and what we could show in a very complicated way, which I won't go into, basically the plant, as the bacteria transitions to this very restricted metabolic state, it stops making its own amino acids, and it needs the plant to provide them. This is really just like, it's part of actually becoming like an organelle. It's shutting down a whole bunch of things because it's energetically more efficient to let the plant do it. And so the plant starts providing a whole bunch of things, which includes amino acids. We could show it was actually branch chain amino acids, because if we manipulated the system so it could still transport just branch chain amino acids, we would now still have a green, happy plant. But anything else, it would be sick. I won't bother with that. And that was just showing that, of course, if we had this mutant, we would restrict the ability of the bacteria to differentiate, because these rhizobia in nodules they undergo endoduplication and they form a chromosome number of around 9 or so. But if we have these bacteroids where we restrict the movement of aliphatic amino acids from the plant, they actually shut down at about 5, and they stop fixing nitrogen particularly effectively. And so that sort of led to this overall sort of summary of the sort of metabolism, where we're shifting a whole bunch of things from the plant. So it's this plant peribacteroid membrane, symbiosome membrane, is controlling nutrient flow into the bacteroid, and the bacteroid in return is providing ammonia out to the plant. This has led us to do a lot of flux analysis to understand carbon metabolism. And I think this is a slightly boring story, even though it's, I think, really important. It's one of the things I worked on for a long time, but most audiences are not interested. But it's simply to show that basically this is what a free-living bacterium would do with 100 units of succinate into its TCA cycle. And then if we go to free-living nitrogen-fixing growth, they have this upregulation of the TCA cycle. But if you're in a bacteroid under very low oxygen, this part of the TCA cycle shuts down. So instead of going from 62 units of flux into citrate, it goes down to 29. And actually, it turns out this is important, because 2-oxoglutarate is the carbon skeleton used to make glutamate and therefore amino acids in assimilation. And bacteroids stop assimilating ammonia. They deliver it to the plant. And this was just really us showing that this is due partly to a very, very low oxygen environment in nodules, and they're able to then control what they do. And this, again, is just showing, I won't bother you with the details, it's simply showing that the bacteroid shuts down ammonia assimilation and delivers that to the plant rather than trying to assimilate it itself. So that was just a recap on how nodules form, and this was just the final bit, if I have time, is something which I think is really started out with a student from Holland who came to do a PhD in my lab, and she was a mathematical modeler, and she modeled what would happen in a real situation if you have a plant where you have good fixers and poor fixers of nitrogen. And she came into my office one day and she said, my model predicts that if you mix a strain which is a really good fixer of nitrogen and a poor fixer of nitrogen, the plant will shut down the poor fixer. And I said, that is absolute rubbish. There's no evidence that's going to happen. And told her to go away. And then I thought about it and I thought, I didn't say it like that. She was a lovely person and we discussed it in detail. But I then said, oh, wait a minute. We do have, and I'm going to prove you wrong, we have a strain which fixes nitrogen at about 30 to 40% of a wild type. If you put that strain on a plant by itself, it will fix nitrogen. You can measure the nitrogen fixation. The plant is a little bit unhappy, but it's better than if you don't give it anything. I said, you mark those two strains, you put them on the plant, and you'll be proved wrong. So she did the experiment, and she proved that I'm an idiot. And it's absolutely remarkable result that basically if you look at, if you take a fixed minus strain, you can't fix any nitrogen. This is the nodule area, all right? They're pretty horrible and small. This is our fixed intermediate strain, the one which I said fixes about 30 to 40%. It's sort of in between, and this is a fixed plus wild type strain. It's beautiful up here and nice and big. The important experiment, what I really want to point out here, is if you take a fixed intermediate strain and compete it either against fixed minus, against itself, or against fixed plus, you'll notice that what happens is when it competes against fixed plus, its size drops dramatically. So the fixed intermediate, if there is this fixed plus strain there, is now much smaller than it would be if it was by itself or against a fixed minus strain. So it is saying, the plant's saying, you're no good, I've got better than you, I'm going to shut you down. And in fact, if you do, a key thing here is really this one here. For example, acetylene reduction is how we measure nitrogen fixation. This is actually showing our fixed intermediate again. And this is how much, if you take the fixed minus, so the fixed intermediate is competing against fixed minus, it fixes nitrogen. If you're against itself, of course, it's fixing nitrogen. Against a fixed plus, it's absolutely fixing no nitrogen. And we could use a series of ways to show that actually the amount of carbon going into the nodules was being shut down when you had better strains present. And this was also just showing that what was happening as well is that if the fixed intermediate competes against the fixed plus, the nodule gets shut down, and actually virtually no bacteria come out of the nodule at 56 days. So in other words, the plant is making a decision, and it's making it after about 12 to 14 days. And the reason is, 12 to 14 days, even a fixed plus nodule hasn't developed yet. So the plant doesn't shut it down till about 14 days. And then it says, how much nitrogen is each individual nodule releasing? And I'm going to compare that against a global nitrogen status signal. And if you don't measure up, it's going to shut you down. And so that's so-called sanctioning, which is essential to the stability of the symbiosis. And I simply point that out because I bet you a lot of things about fungal biology are going to have the same sort of thing happening. It's not going to be exactly like this, but about selection between strains of fungi or other bacteria. There'll have to be some sort of mechanism like this. And this was just us showing that the selection occurs at the nodule level, not at the single cell level. It's just showing this is where we force two strains into one nodule. You can do it if you use very high infection. And if you force a fixed plus and a fixed minus in together, it doesn't shut down the cells of the fixed minus, it shuts down the whole nodule. It assesses the nitrogen output from a nodule. And then I'm going to stop. Let me see if Annette's disappeared. This is Tom, who's done most of that sanctioning work, and Mari, who did a lot of that as well. There's Haley and Raphael, who did a lot of the that sort of in-seq analysis, looking through the population over time. And I'll stop there because I've probably talked too long anyway. So thank you for your attention. Yeah?
I'm curious if you have any sense for how applicable some of the work you've done here in rhizobia is to actinolysal, like nitrogen fixers. Do you expect that like some of those mechanisms are going to be similar, or do you think it could be completely different?
I think it's a very, very, very good question. I think a lot of it will be similar, but there'll be other differences. And there are even differences within the legume types, because there's another type of legume nodule, it's called a determinate nodule, where the bacteroids do not terminally differentiate, and so there'll always be, and so we've just done a big analysis of that, and there are a whole bunch of different things going on there. I mean, I think that the point is you have to stabilize the symbiosis. So I think it doesn't matter whether it's actinorhizals, whether it's Frankia, or whether it's rhizobium in there. A lot of the same things are happening. And I guess the key thing is a plant can't determine in advance how much nitrogen any infecting strain is going to give it. It can't detect that because they don't fix nitrogen, Frankia or rhizobium, until they're inside the mature nodule. So this is the idea that to stabilize the symbiosis, the plant has to sanction the nodules. It can't select the partner, although there is partner selection in terms of other things. You know, do you have the right cell surface? Do you produce nod factors and so forth? Yeah. But I think it'll be really, really very similar. There'll be a lot of similarities, maybe different mechanisms for the same processes. Yeah? That's a really, really good question. And if you think about it, in a natural system, it probably depends an awful lot on how much of that particular legume that's compatible has been in that soil. Because what's happening is, of course, there's a positive selection through this sanctioning of pore fixing nodules. The frequency, the population frequency of the good bugs, if you like, are going up. But of course, that's in terms of over time. This is other bugs may be better at surviving in the soil, and so there's a dynamic in there all the time between good fixing bacteria and poor fixing bacteria, and what has a selective advantage at different times. But the key thing is, at least in pea nodules and many other so-called indeterminate nodules, it's the number of the good fixing bacteria that come out of the nodule which are going to determine, at least immediately afterwards, that a boost in population frequency. But actually, it's an ecological dynamic, isn't it, between all the other edaphic factors and the survival in soil and so forth, which will change the composition of the population. And probably I probably should have given more of a lecture on that stuff than the molecular stuff to this audience. But anyway, it's a great question, though. Yeah. Yeah.
So in your TNC, you recover the bacteria from nodules and then sequence them.
Yes.
You're not going to recover any gene that's important after differentiation, because those bacteria will not be viable after.
It depends. It depends because, of course, it's true. The differentiation is the final stage. It is the final stage. So it's true. If you're required for differentiation, there's no further selection after that. But remember, those final differentiated bacteria are terminally differentiated. And the really super interesting thing is, if you go to bean nodules like Phaseolus beans, it's the bacteroids which de-differentiate, and we've just redone all this analysis, and there's a whole bunch of new genes which are required to be able for the bacteria to de-differentiate back into free-living bacteria. And a whole bunch of things like iron mobilization genes, and genes which are essential in mitochondrial development. And that's really exciting because mitochondria are rhizobia. And so there's a whole bunch of exciting possibilities there. I'm going off the question, but you're correct, yeah. Yeah, that's true. Great. Thank you very much.
Great. So, all right, everybody, we're going to get started again. So for the last portion of the morning session, going up to lunch, we're going to have 2 more talks. And to introduce the first speaker, we've got a PhD student in OAB, Calvin Heslop.
All right, it is my great pleasure to introduce our next speaker, Dr. Michelle Wong. Michelle is an assistant professor at the Department of Ecology and Evolutionary Biology at Yale University. Prior to starting her position at Yale, Michelle did her undergraduate degrees at Berkeley, where she received a BS in molecular evolutionary biology and a BA in sociology. She then spent time as a lab technician and then as an environmental scientist with EPA, before doing her doctoral work at Cornell University. She then took a postdoctoral fellowship at the Carey Institute for Ecosystem Sciences, where she worked with Sarah Batterman. Michelle's work focuses on the biogeochemical cycling, primarily in tropical forests, and is probably best known for her work on the molybdenum cycle and how nutrients like molybdenum and phosphorus impact nitrogen fixation in tropical forests. And I think I can speak for all of us by saying molybdenum and phosphorus probably in all of our top 5 best chemical elements. Today, Michelle is going to span the tropical-temperate divide as she tells us about plant microbial interactions in response to nutrient limitation in tropical and temperate forests. Please help me in welcoming Dr. Michelle Wong.
Oh, okay. Can everyone hear me? Thanks so much to Calvin for the introduction and for Ben for organizing this awesome symposium. It's been really fun to hear all the talks this morning. And I'm going to zoom out and go a little bit more to the ecosystem scale, so no omics work or any genes. And I'm going to be talking about nutrient limitation. And I think most of us know that terrestrial ecosystems play this really critical role in the global carbon cycle, taking up about a third of the carbon that we emit on average. And there's big questions as to what extent this land carbon sink is going to continue into the future, or if eventually we'll see more resource limitation by things such as nutrients. And this is just a map of projected nutrient limitation spanning nitrogen and phosphorus. And you can really see that quite a bit of, most of the terrestrial ecosystem has experienced some, or projected to experience some nutrient limitation, although there's quite a bit of spatial variation and also temporal variation that we'll talk about today. And I think all of us in this room also know that plants can use different strategies to actually address nutrient limitation. So they can use nutrients more efficiently or produce fine roots in response to acquire more nutrients, or release enzymes that can break down organic matter. And then they can also rely on some partnerships or symbioses with different microbes, such as nitrogen-fixing bacteria, mycorrhizal fungi, and then they can also recruit in microbes through the release of root exudates and also modify their soil environment. And I think one of the big questions that a few of us in this room are motivated by is to understand to what extent can these plant-microbe interactions increase or decrease that land carbon sink into the future. And so this is a modeling projection of a land carbon sink with and without incorporation of nutrient cycling into the future. And you can see that when models incorporate nitrogen or phosphorus limitation, they find that NPP actually decreases, and that the land carbon sink can actually be a source. And I think as we start to represent more plant microbial interactions in these models, it's going to be curious to see if whether the incorporation of some of the mechanisms is actually going to minimize nutrient limitation or potentially even release potentially more soil carbon if we switch over different functional types of mycorrhizal associations, for example. So still a really big question. And so today I'm going to be asking if a better understanding of plant microbe interactions will help us better predict how terrestrial ecosystems will respond to global change. And I think the symposium title was Root Microbe Interactions in a Changing World. And traditionally, when we think of nutrient limitation, we expect that it's going to increase with elevated CO2 as more nutrients are bound up in plant tissues, or just due to increasing land use change, which often we lose nutrients in the process. And you might expect that in response to nutrient limitation, you get declines in biomass. But we also know that plants can rely on some of these strategies, and that might minimize nutrient limitation because of increased carbon allocation to those symbionts or whatever to actually minimize what we'd expect nutrient limitation to look like. But we also know that tree species can actually turn over, and so you might have, in different soil fertility conditions, you might have the mortality of different species, and then also different species recruiting in. So your community composition might look very different, whereas the biomass might actually look the same. And we see this across nutrient gradients all over terrestrial ecosystems. And maybe we can start to predict which species are going to do better under that community turnover because of their below-ground strategies. And maybe they can map onto simple functional groups such as AM or EM or unfixing species. So still really big questions. So the bigger questions that I'm going to be asking today are how consistent are these plant microbe functional groups like N-fixers? Do N-fixers all behave very similarly across ecosystems, right? And then how predictable and flexible are actually some of these strategies, such as investment in mycorrhizae or into N-fixation, and how can we predict that based on nutrient availability? And then finally, a combination of asking if those plant microbe functional groups actually are predictably responsive to nutrient availability, which is still a really big question. So I'm going to talk mostly, I just started at Yale, so I don't have anyone in my lab right now, but so I'm going to be talking about my postdoctoral work and then my PhD work. And a lot of folks were involved with that in 3 different regions. Okay, so moving on to that first question, I'm going to specifically focus on N-fixation, and I think most of us in this room know that at least the dominant thought is that most N-fixation occurs in tropical forests and savannas. This is one of the modeling projections that really shows this hotspot of N-fixation across the tropics, especially within the Amazon basin. And the Amazon basin, especially in the lower Amazon, is part of this arc of deforestation where there's quite a bit of, or a lot of land use change occurring. And so a lot of forests are being cut down and converted to either pastures or to croplands. And this really impacts the downstream water cycle because as those trees are cut down, they're no longer transpiring water. and become really quite flammable. And when you mix that with agricultural forest fires, these fires can spread, especially in drought years. So I think all of us remember when Amazon forest fires made it onto the news in 2020. They're actually projected to get a lot worse this year. I think it might not make it on the news, but that's just the state of how tropical forest fires are looking these days. And even though tropical forests are not really thought to be nitrogen limited, they can be after a fire because nitrogen tends to volatilize at lower temperatures, which can actually push some of these ecosystems maybe into short-term nitrogen limitations. So in a previous study within the Amazon, they found, Kaufman et al. Found that about half to 60% of nitrogen was lost, and then about 7 to 30% of the phosphorus was lost. And so there's this potential for these nitrogen-fixing trees to play a critical role in that short ecosystem recovery time. And so we know that from some of these secondary forest experiments that there's higher symbiotic N fixation, also higher free-living N fixation in some of those recovering forests, as well as a higher abundance of nitrogen-fixing trees, especially in dry tropical forest sites, which are indicated here by the orange color. So quite a bit of abundance of nitrogen-fixing trees that tends to decline as these forests mature. So a lot of evidence pointing that they might play some functional role. And today I'm going to be taking us to a part of the southeastern Amazon where they actually had conducted, or my collaborators conducted, this large-scale fire experiment. And so these forests are 50-hectare plots that were burned. At least one of the treatments, it was burned 3 times every 3 years from 2004 to 2010. And because we have information on the leaf area index and litter fall and tree inventory info, we can track some of the behavior of some of those trees, as well as the nitrogen budgets. And I'll just quickly walk through this. So we tracked the wood nitrogen budget, which declined by about 50%. And we're not really seeing recovery, at least as of 2016. We haven't really started to see a recovery in that wood nitrogen pool yet. And then in the leaf nitrogen pool, we're actually starting to see a recovery at about 5 kilograms of nitrogen per hectare per year, which I'll contextualize in a little bit. And so we also wanted to see if N fixation played a role in building up some of those new nitrogen pools. And then the other question we want to ask is if the abundance of N fixing trees actually affects forest growth. So we might expect that the higher or more abundance of N fixing trees you have, the faster your forest might recover. And then we also might expect that, again, as forest age increases and forests are recovering, you might see a decline in that initial abundance of those N-fixing trees. Okay, so what did we find? Well, overall, at least in our experiment, we didn't find that the abundance of N-fixing trees based on their basal area actually changed overall basal area, had any effect on total forest recovery, and no effect also on N-fix or non-fixing tree basal area as well. But we did find this really large increase in the actual abundance of those N-fixing trees, so about 1% to about 10% in those burned forests. So really rapid growth, at least, of those trees. Okay, so what are they actually doing? So we measured N-fixation between 2016 and then 2017. And so asking if N-fixation actually increases post-fire to replace some of those losses. And we measured both free-living and symbiotic N-fixation, took over 1,000 cores, and then we also cored doing a systematic approach. So we did a gridded sampling approach across the forest, knowing that some trees are going to spread their roots tens of meters away, and then also around the base of different N-fixing trees as well. And then we also measured free-living N-fixation in the litter and the soil with a settling reduction assay, and then we paired it with 15 N2 incubation. And what we found was that even though symbiotic N-fixation increased by a little bit in those burn plots, they weren't enough to actually make up basically those nitrogen losses in the forest fires. And when we incorporated free-living N-fixation in there as well, we still found less than 1 kilogram of nitrogen per hectare per year fixed in these forests. And these are really, really low estimates. And just to compare, through Ben's study and Joy's, who's in this room, study as well, our sites, basically even in those burn plots, those N-fixation rates were incredibly low compared to a lot of these other sites in the tropics. So I think one of the big questions is what explains a lot of this variation across the tropics, right? Is it biogeography? Is it just soil nutrient availability? What is it? And one of our hypotheses for the reason why we didn't see this huge increase in N-fixation, despite this increase in abundance of N-fixing trees, was due to the soil type, which was super weathered oxasols. And even within the top 4 meters, you can find about 220 kilograms of extractable nitrate per hectare, and because of potentially anion exchange capacity. And just to show you how weathered these soils are, this is Santorum pulling out a soil auger from the ground that actually almost cores like butter. It's incredible how easy it is to get a soil auger in the ground and out of the ground. But yeah, this is going to 10 meters. And I also almost toppled over once trying to actually carry the auger. So then I think the next question is how common is it? How common is this, right? So are we just not measuring N fixation in enough representative ecosystems? And this is just a really coarse mapping exercise of similar soil types that are topographically stable. And about 40% of the tropics might be similar to some of these sites in the lower Amazon. And they might also have quite a bit of anion exchange capacity, and maybe that could explain patterns of N fixers. Who knows, right? Here I have plotted or mapped on different nitrogen fixation studies, and I think maybe taking some of these approaches to think about the underlying soils and biogeography is going to help us understand our biases and how we actually know about how N fixers function. So in summary, N fixing tree abundance, at least at our sites, didn't affect forest growth, and they also didn't increase post-fire to replace those nitrogen losses. And I think to really understand N fixers as a functional group, we need to improve our understanding of spatial variation in biogeography and in soil variation. So that's the first takeaway point. Okay, so next we're asking how flexible and predictable are some of these plant microbe interactions across gradients in nutrient supply. And I'm going to specifically focus on plant and microbial phosphatase activity, and also asking if plants adjust investment in mycorrhizal fungi in response to changing nutrient availability and changing plant demand. And I'm going to focus on a clear-cut secondary successional forest in the tropics. I think a lot of us know that about 50 to 60% of tropical forests are in some stage of recovery at this point. There's really rapid rates of land use change. And we might expect that there's really rapid regrowth early on as trees are trying to capitalize on this high light environment, but that these growth rates slow down, and so you'd expect the same for nutrients accumulating in biomass. So there's quite a bit of external nutrients required early on in succession. And in addition to the strength of nutrient limitation, we might also expect that the type of nutrient limitation may shift a little bit. And that's because phosphorus tends to stick around in soils after clear-cutting events, whereas nitrogen is really mobile. You see a lot of nitrate export in streams after forest cutting events, and then also more denitrification. But that nitrogen usually can come and be replaced theoretically by N-fixing trees. So you got this potential shift from early nitrogen limitation to later phosphorus limitation, or maybe some other nutrient like calcium, in addition to the change in the strength of nutrient limitation. So I'm going to be focusing on phosphatase. And if you don't know what that is, it's basically an enzyme that cleaves off phosphate groups that plants or microbes can later take up. And both plants and microbes can actually produce these enzymes. And then I think everyone knows what the important role that mycorrhizal fungi play, but theoretically they're exchanging carbon for some access to increased resources. And I'm going to talk about my mycorrhizal colonization specifically today, and then the system that we're working actually is in Panama, and there's my collaborator set up a multifactorial fertilization experiment across 4 forest age classes that started around age clear-cut into pasture, and then also 10-year-old forests, 30-year-old forests, and then mature forests, at least 300 or 600 years old. And the plots were replicated about 4 or 5 times depending on the forest age. And so we're going to look at, I'll show you some of the results of the root and soil phosphomonoesterase activity, and then also mycorrhizal colonization responses. And I think some of us who've done root staining might not know it's not the best proxy for investment into mycorrhizal fungi, and I'd love to hear more feedback on some other ways we can start to measure besides doing carbon tracing into seed leaks. Okay, so we might expect that across forest age, root phosphatase activity might increase as nutrients become more limiting across forest age, and then decline again in those mature forests as there's less mining for new nutrients. And that with nitrogen, maybe because phosphatase has a nitrogen cost, about 15 to 20% of these enzymes are made up of nitrogen, there might be some stimulation of phosphatase activity early on in succession, and then perhaps a suppression by phosphorus only later on in succession. Okay, so I'm going to show you these results 4 years after fertilization, so the forest ages have changed a little bit, and I'll show you the results just for the root scale. So this is activity per gram of root, for example. Okay, so within the control forests, we actually find this slight increase across forest age and then a slight decline again in those mature forests. Okay, next I'm going to show you the phosphatase activity results as we scale them to the total fine root biomass across the system. And basically we find the same patterns. We see this increase across forest age as we expect more phosphorus limitation, and then a slight decline again in the mature forests. And then I'll just show you the soil phosphatase activity. And this was kind of a sanity check for us just to see if it's really variation in underlying soil. Like, are we seeing this pattern because of the underlying soils? And what's really interesting is that monoesterase activity just within the free living soil bacteria increases across forest stage. So there's some decoupling occurring between the plants and the microbes. And so this is just on the per gram of root, again, so just scale to the root. And we actually see a stimulation of root phosphatase activity like we predicted in response to nitrogen in those younger forests where we expected nitrogen limitation. But then no stimulation of phosphatase activity in those older forests. So no nitrogen limitation, at least in those older forest age classes. And then no suppression of phosphatase also by phosphorus in those younger forest age classes, but then a suppression by phosphorus in those older forest age classes. So kind of these predictable responses, at least to those plant root enzymes. And then for when we scale up those results, we actually lose that nitrogen stimulation effect, but then we still get that phosphorus suppression effect, particularly in those older forest age classes. So they definitely seem to be responding to their environment, and then also across forest age in response to nutrient demand. And then for the soil phosphomonoesterase results, we basically see a suppression, and more so in those older forest age classes, and then a slight stimulation in the later on older forest age classes to nitrogen. So definitely a decoupling between those plants and microbes. Okay, so in response, at least for the root phosphatase activity, we kind of basically saw a lot of the responses that we expected, at least on that per root basis. Okay. So for mycorrhizal colonization, we expected to find a decline in colonization over time potentially because of like less mining for nutrients in those mature forests, and maybe a slight suppression or a slight stimulation in response to nutrients because the fungi can also be nutrient limited early on in succession, and then perhaps a suppression later on in succession in response to nutrients. Okay, so what did we find? Well, across forest age, we actually didn't see any changes in mycorrhizal colonization across forest age like we expected to find, and then really no response to nutrients in those younger forest age classes, and then a slight suppression in response to nutrients only in those mature forest age classes. So some interaction between nutrient availability and forest age where those mature forests were responding. So in summary, I think we didn't, at least at our sites, we didn't really see this decline or stimulation, maybe some suppression only in those older forest age classes. And I think this is another question that we have is how representative are these patterns of other tropical forests? So we did a meta-analysis of glasshouse seedling experiments and also field experiments of root phosphatase and then of mycorrhizal colonization responses to nutrient availability. And at least root phosphatase, just like in our experiment, seemed to be relatively responsive to phosphorus and then potentially to nitrogen, maybe depending on the variation in nitrogen availability across a lot of these sites. Whereas for mycorrhizal colonization, we actually had, in the parentheses, we have the different number of observations, and we really didn't see any responses to nutrients that were significant except a slight suppression in response to nitrogen. So I think this really, I mean, all this really is saying is that we're not seeing really consistent responses of colonization specifically. So I think this is, if we're interested in looking at carbon investment into mycorrhizae, like we have to start thinking about some perhaps some better methods, or perhaps this is a reality and not an artifact. And then we also looked at some studies that had measured colonization across forest age too. And on average, I mean, we did find a slight decline of colonization across forest age, but there was a lot of variation within sites as well. And there's some hypotheses that perhaps in areas that receive like really high disturbance rates, they might actually be more beneficial or better off being less mycorrhizal early on and rely maybe on, instead of pioneer species being more mycorrhizal. So maybe disturbance history could help explain some of these patterns, but again. Yeah, so in summary, we found really predictable responses of root phosphatase in response to changing nutrient availability and demand, but we didn't really see this for mycorrhizal colonization specifically. So how can we start to better measure investment in fungi? And then the other question is really what is the cost of these strategies, right? So if we're trying to model some of these, which I think maybe Eddie will talk about today, if we're trying to incorporate some of these plant strategies in response to nutrient limitation, the carbon cost might really set an upper bound on whether or not we're going to see alleviation of nutrient limitation. Okay, so the last component of this is really trying to get at plant microbe functional groups and if they respond to nutrient availability in predictable ways. And specifically, the question here is asking if N-fixing trees actually play a role in accelerating phosphorus cycling in temperate forests. And the reason for this is that typically we think of temperate forests as historically being nitrogen limited, and we also know that they've experienced quite a bit of nitrogen deposition, especially in the eastern continental US. And there, at least between 2015 and 2018, there was some evidence that maybe we're seeing a shift towards phosphorus limitation or co-limitation with nitrogen. So one observation was from the stoichiometry of different forest leaves, and then also in the Melnahe experiments up at Bartlett experimental forests, we're actually starting to see some growth responses to phosphorus fertilization, at least of the trees. And then I think the newest update from Ruth Unai is that actually maybe there's more co-limitation by nitrogen and phosphorus, but so basically changing biogeochemistry in eastern forests. And what's also really interesting is that black locust, or Robinia pseudocation, is really the dominant N-fixing tree in eastern forests, as indicated by this purple color from an analysis of FIA data. So this overlap of historical N deposition and also a high abundance of black locust trees, at least of all of these N-fixing species in eastern forests. And I think Annika Stocconi's work showed that about Robinia is responsible for about 64% of all symbiotically fixed nitrogen within the continental US. And what's really interesting is that you might expect that under N deposition, N-fixers would actually do more poorly. But what's really interesting is that from another analysis of FIA data is that black locust had one of the highest growth responses to N deposition, and then also one of the highest survival rates in response to N deposition. So kind of the opposite of what we would expect, because you might expect that nitrogen-fixing trees would do better under nitrogen-limiting conditions, right? And so maybe it's that they're offsetting some of that carbon cost that normally goes towards nitrogen fixation elsewhere, or either towards seed reproduction or growth. And one of our hypotheses as the reason why it could be also doing well under N deposition is if it accelerates, it can accelerate phosphorus cycling. And the hypothesis there was that N-fixers have historically always been able to alleviate some of their nitrogen limitations, so that perhaps they have strategies that are more focused also on acquiring phosphorus. So maybe N-fixers are just maybe more phosphorus limited than some of these other non-fixers, like aruscular or ectomycorrhizal tree species. And really the main pools of phosphorus in the soil are either mineral forms of phosphorus, organic forms of phosphorus, which are probably actually the most dominant sources that get recycled, and also forms of phosphorus that are sorbed onto soil particles like iron and aluminum oxides. And all of these forms of phosphorus can actually go into solution to be taken up by plants or by microbes. But plants can also modify their soil environment as well. So there's some evidence that under nitrogen fixers specifically, there's lower soil pH because of enhanced nitrification rates and more nitrate accumulation. So there, at least from plantation experiments, they found lower soil pH below nitrogen fixers. And that can potentially enhance rates of mineral rock weathering, ? And perhaps there's some evidence of nitrogen fixers being more mycorrhizal. And then also one of the big hypotheses is that because phosphatase enzymes have quite a high nitrogen cost, that some of that fixed nitrogen can actually go towards phosphatase. And that's one of the hypotheses that maybe explains why N-fixers are really abundant in the tropics specifically. So we wanted to actually compare some of those phosphorus strategies among dominant fixers and non-fixers across eastern forests using this experimental and then an observational approach. So for the experimental approach, we grew 7 different species. Sugar maple, honey locust, cherry species, and then 3 ectomycorrhizal dominated species, and then 4 arbuscular mycorrhizal species. And then we grew them across 3 levels of nitrogen, which I won't really show a lot of the results for today. And then we actually grew them in a phosphorus-free medium, and then within the pots we added litter bags for organic sources of phosphorus, and then bags of apatite for rock mineral sources of phosphorus to see basically how good these plants were at getting some of these phosphorus sources. And then we replicated the experiment and grew them throughout 18 weeks. So you can really see, especially black locust grew quite a bit within those 18 weeks. Okay, so the first, and then I'll talk about specifically about those different results. And yeah, I think Philip made a joke about picking nodules. I think some of these plants took us like 10 hours to pick all the nodules off of, actually. So they were inoculated a little too well. Okay, so the first result I'll show you is just the phosphorus uptake in the plant tissue. And what we did find was that black locust, or Robinia pseudacacia, like we hypothesized, did take up the most phosphorus throughout that course of that 18-week experiment. Black birch also did a really good job as well. And then I grouped some of these species by either N-fixers or arbuscular ectomycorrhizal tree species. And then we didn't really see, because we had the apatite bags buried in those pots, we could measure mass loss change over time. I mean, this was a really short period within 18 weeks to look at mass loss, but really no differences, at least within the seedling experiment. But what was really interesting is that we did have a no plant control, and actually the mass loss was much higher in the no plant control, perhaps due to maybe more like moist conditions, but also higher microbial activity. And there also seemed to be a response to N availability too. So a lot of that breakdown of that appetite was probably microbially induced, which is interesting. And then we also looked at mycorrhizal colonization. I think there's going to be a lot of differences because we used plant root colonization versus just tip colonization for ectomycorrhizal tree species, but this really varied a lot depending on the species you were looking at. So no evidence of any differences in colonization, at least. But what was really interesting was, at least on the per gram of root basis, we did find much higher phosphatase activity for Robinia. And then when we scaled that up to fine root biomass too, because the Robinia were a lot larger, they did produce a lot, orders of magnitude higher phosphatase activity. So that could have been perhaps one of the reasons why they were able to get more phosphorus accumulated into their plant tissues. And so this is just an artificial greenhouse experiment. So we also wanted to compare some of these results with mature forests, or with mature trees across some of these different forest sites. And to do this, we used 6 of the 7 species that we had in the greenhouse, and then we traced roots, which I think, maybe everyone, or maybe half the room is actually traced. Has anyone not traced roots before? Or? Okay, a few people. So I'll just show an example of how tedious this can be. So this was a white pine that we had. And this is like the fine root tip that we were trying to collect, and I think it took us over 10 hours, maybe 8 hours to actually get some of those fine roots. So it's really labor-intensive work, and there's probably some other approaches we can use to get at roots at the community level, and then go with an omics approach. But at least if you want to know the root that you're sampling from the tree, this is oftentimes what we do to get there. And we also did collect rhizosphere soils, so if anyone wants to analyze them, we just put them in a -80 and didn't do anything with them, but we have those, because it was so valuable to actually get those soils. And then because this was in the forest site, we actually were able to bury those apatite bags for about a year in the field, and we actually did find slightly higher mass loss, at least for sugar maple, but I think it's because they were going after the calcium in apatite, and they're really calcium-demanding species. But basically no big differences in rock weathering. And I think this could be due to the fact that we didn't actually see differences in soil pH because we were working in really mixed stands. And also the black locust, they really are like more early successional species that do well on edges. They're not really great in mature forest sites. So all the black locusts that we sampled were not the happiest. And then we definitely found higher mycorrhizal colonization rates in the field as well, and then lower rates for the ectomycorrhizal tree species because we were looking at root tips. And I'll show you the phosphatase results. So because those mature Robinia were in the mixed field forest plots were not as happy, we also sampled some of the saplings, and we did find slightly higher phosphatase activity rates, which was consistent with our greenhouse experiment, but not to the same magnitude that we saw in the greenhouse experiment. So basically, our main hypothesis or what we really found to support that higher phosphorus accumulation in their plant tissues was due to potentially the phosphatase activity that was breaking down some of that organic phosphorus. So in summary, we found that Robinia at least seems to accelerate organic phosphorus cycling, this nitrogen-fixing species, at least for those younger forest ages. Then I think one of the big questions is if N-fixers as a group, again, are functionally similar, and when and where are groupings such as N-fixers versus ectomycorrhizal associated versus arbuscular mycorrhizal associated enough to explain nutrient acquisition patterns, because we didn't really see a breakdown, at least of the 7 species that we studied here, but I think as we try to represent more functional groups into models, like when and where can we actually get those breakdowns? So to what extent can plant-microbe interactions actually increase or decrease the land carbon sink? I think just the presence of N fixers alone doesn't always mean that they're fixing nitrogen, and there's quite a bit of spatial heterogeneity just in the biogeography of the tree species and probably the microbes themselves, as well as soil variation. Okay, we did find some evidence that plants can kind of upregulate some of these nutrient strategies, and I think I've been saying this for like most of the talk, but we need a better way to measure investment in mycorrhizal fungi, so I'd love to chat with people about that. And yeah, I think moving forward, how do we better represent those functional groups or guilds? How can we start to better understand the drivers of spatial heterogeneity? How can we start to better get at plant community responses to nutrient limitation rather than just individual species? And how do nutrients interact with other drivers? So we know that there's going to be mixed effects with drought and warming, for example. And then also coupling above and below ground carbon dynamics, which I didn't really get a chance to talk about today. And with that, I'd like to thank the funding sources and a lot of other folks who helped with that work. And I'll take any questions.
Started to unpack a bit of disturbance in there and how time since disturbance might affect that. And I wonder if you could just talk a bit more about that.
Yeah, so I think all of the, we had to classify, because that was a review, and so we classified, I guess, all of the zero as like freshly disturbed ecosystems. I think the extent of disturbance I didn't account for, so some of them could have been like full clear cutting and some of them could have been like post-fire. But I think, or if you're talking, are you asking about the variation across the studies, or?
Yeah, and just like I just think about, disturbance is kind of systems experience it all the time naturally and anthropogenically, and I think it's just really interesting to think about how time since disturbance might affect like what we observe as a thing in that moment when we're not thinking about disturbance. I like that you kind of started thinking about that and just wanted to hear more of your thoughts.
Yeah. I think in that that's a great point. I think in that x-axis too, like what we wanted to account for was like looking at if I guess one of the questions kind of buried within that is if the pioneer species are inherently more mycorrhizal than later successional species. And I think maybe I didn't explain this really well, but I think there's questions as to when pioneer species being very mycorrhizal is actually beneficial versus not. And that depends on like if in some of those sites like Puerto Rico that have experienced a lot of disturbance, it might not be beneficial for those pioneer species to actually be super mycorrhizal because they might not have access to their symbiotic pool. So that was kind of the theory as to why you might expect colonization potentially to increase across succession as well. So lots of things to think about.
Kind of along those lines, did you see a difference between disturbances like fires and then something like a logging event? Did that affect how the succession?
Yeah, that's a great question. I don't think I took, I think I took notes in the studies, but I don't remember, it didn't stick out to me which studies actually found differences. But I think the site, I think Bene Bachelat, they actually found this, they were the ones that found the increase in colonization across succession, which they did attribute to more extreme disturbance like hurricane events, I guess. Well, that's not as much.
Do the N fixers in the tropics accelerate phosphorus cycling? Because I'm trying to understand like why there's so many N fixers in the tropics, but a lot of nitrogen.
Yeah, that's a really great question. So I think there's still quite a bit of support that N fixers have higher phosphatase activity rates relative to non-fixers, but it's not due to the rate of N fixation. So I think the original theory that like Ben Holton had proposed was that if the more fixation you have, the more phosphatase you produce, and that relationship at least breaks down within species. But I think there's some press work from another group at McGill, Fiona Soper's group, that shows that if you actually incorporate more species in there, you still do get that relationship. But I think at least from some of the studies we've seen, and then also from fertilization experiments of seedlings, we see that N fixers respond more to phosphorus than non-fixers, at least of the seedling stage. But yeah, great question. Colin?
Awesome work. I wonder what your experience is of like water availability in those burned forests. I think a lot about nitrogen fixation in the tropics. Is that, and I realize like it's the Amazon, right? Like it's famous for being wet. And so, what do you see there?
That's a great point, and I think that's why we're actually seeing this increase in abundance of unfixing trees, and not related to their fixation specifically, but potentially due to drought tolerance. Or I mean, maybe, I don't know. I think it's really driven by just a few species, and I don't think we know enough, at least of those sites, about their drought strategies. But that's like my hypothesis as to why they're increasing.
You showed a slide that the rubidium was responding more than the other species to nitrogen deposition.
Mm-hmm.
What was the nodule density in response to that nitrogen deposition? Did it have fewer nodules, or was there some sort of compensation?
Yeah, really great question. So that data was from FIA data, which I don't think they've actually explored nodule biomass, but what's really interesting is that Nina Wurzburger has resampled some sites at, I think it's Coweta, where they basically find, at least when they compare it to like a study that was conducted decades ago, way less end fixation due to historical end deposition. Or that's what they think. I mean, they use different methods, and it was someone else who was like analyzing the nodule biomass, for example, but the hypothesis was that end deposition has caused declines in fixation too. So, great.
To keep us on time.
Okay. Sorry.
Thank you, Michelle.
Okay, it's my great pleasure to introduce Dr. Adriana Corrales, who is a leading expert on ectomycorrhizal ecology in the neotropics. Adriana's work focuses on integrating fungal community dynamics with ecosystem processes, primarily working on how ectomycorrhizal partnerships impact the function of tropical montane forests. Dr. Corrales is the leader of expeditions strategy and planning for SPUN, the Society for the Protection of Underground Networks. Prior to her current role, Dr. Corrales was an assistant professor at Universidad del Rosario in Bogotá, Colombia. Earlier in her academic career, Adriana did a master's at Universidad Nacional de Colombia in Medellin, and then went on to do a PhD at the University of Illinois Urbana-Champaign, where we overlapped briefly. And then she received the highly competitive Ewell postdoc fellowship at the University of Florida. I hear that Adriana's current work involves travel to lots of amazing places, such as the Atacama Desert. So, and there she works to characterize the fungal diversity in such understudied ecosystems. So, if you're interested in traveling to the ends of the earth to study fungi, find Adriana at one of the breaks. So please join me in welcoming Dr. Adriana Corrales.
Wow, thank you so much for that very nice introduction. I'm very, very happy to be here, sharing some of my research here. Yes, I'm working right now with SPUN. Most of what I'm going to show today, I did it with Universidad del Rosario, with many undergraduate students mostly, over there in Colombia. Okay. That's it. Very good. Up. Thank you. Okay, perfect. So Michelle did an excellent job. I'll introduce in tropical forests, so I'm gonna go fast in this part. But when we usually think about tropical forests, we think about these beautiful ecosystems, these beautiful animals that we have over there. But in reality, many of them look like this. A lot of fire, crops, and cattle, which is alarming. It's alarming. Deforestation is happening at alarming rates. There are many, many fronts of deforestation. And sometimes when you don't see deforestation, it's because there are no forests left. For example, in Colombia, what you see in green here are the forest cover. The other parts are no forest. Most of it is deforestation. A little bit of it is prairie or grasslands, but we deforest over 150,000 hectares per year or more. And that trend increases and decreases depending on the year, but more or less. So my work focused on the Andean region, which is the mountains of Colombia. If you see, a lot of it is not green. It used to be green, but it has been deforested. However, it's the second most deforested area with almost 17% of deforestation in the country. So even though it's really deforested, deforestation keeps happening. These are really, really fragmented ecosystems, really deforested, basically. And of course, this deforestation matters for the underground, right? For example, fires reduce fungal biomass by up to 96%, and globally, 90% of the soils are expected to be degraded by 2050. And I'm just going to give you a little bit of introduction about the SPAWN here, because I hear people are curious. And Colin that is here is one of the co-founders. Basically, we want to protect the underground, specifically in mycorrhizal fungi. We want to do very detailed maps of mycorrhizal biodiversity all over the world. We want to protect those mycorrhizal fungi, trying to include them in reforestation and also into policy for conservation, and also trying to find where they are good for to protect climate. And we want to do that with machine learning algorithms for very detailed maps of that biodiversity, and also by creating a very large network of mycorrhizal ecologists or mycologists all around the world that could help us to achieve these very ambitious goals. So going back to the tropics, these tropical phagelis live in that very deforested mountains of Colombia, and they are not only in Colombia, but all over the world. These mountains look pretty similar, and they are in a very special belt called the cloud forest, where we have a lot of foggy areas, and this fog, it has been shown is disappearing. This belt is disappearing due to climate change. And these phagelis are very special. They form monodominant forests. There are ecosystem engineers, and they are in these groups. They are not very diverse. We only have Fagaceae and Juglandaceae, mostly from ectomycorrhizal associations, Quercus, Trigonovalanus excelsa in the Fagaceae, and Oremunia, Alpharoa, and Juglans that are in the Juglandaceae family. Why I'm showing this? Because these are all considered relict plant species. They used to be organisms that were widely diverse and widely dispersed all over the northern hemisphere mostly. And they are reduced to very, very specific locations. They have very small populations, and therefore they are very fragile. And if you think they are very fragile and then put that in the context of deforestation, fragmentation, and climate change, they could very easily disappear. Also, because they act as inoculum islands of ectomycorrhizal fungi. And I'm going to tell you why in a second. Well, because most tropical ecosystems are dominated by arbuscular mycorrhizae. I'll show you in a little bit. So this is, for example, one of the species I'm going to show you a little bit of research on. Trigonovalanus is an endangered species. Juglans neotropica, another endangered species I'm working on. And many in the Juglandaceae, 20%. There are only 60 species globally, and 20% of those are in the IUCN either as vulnerable or endangered. And most of them are in the tropics and subtropics. So this is a risk. They are very risk. So, for example, this is one of them. This is a least concerned species. Quercus humboldti is, if you see, distributed exactly in that very deforested area of Colombia in the mountains. And something really interesting about it, it believes that its center of origin was in Mexico or somewhere in North America. And it migrated through Central America until Colombia. In Colombia, we only have one species of Quercus, and it's the southernmost distribution of this genus in the American continent. And it's very interesting that the hypothesis is that this Quercus migrated down the continent with its symbiotic fungi. We are trying to demonstrate this is a huge task, especially because most of the species are undescribed. We described this new species, Rusula floriformis, that we found in Panama and in Colombia, and we found actually that they, they are really very, very close, and they are found in these very divergent populations because in the middle we have the Darien, which is lowland forest. So Quercus is not present there. So we really think this is happening, that they are probably starting to speciate, and they all came from the northern North America. We found this in another new species, Rusula ferruginea. We always find these very, very close subspecies that are one in Panama and one in Colombia. That's all we have so far. We're working in more Rusula species to try to see what happens. But then what are the global patterns of ECM symbiosis? Then if you see the red areas are arbuscular mycorrhizae, the blue areas are ectomycorrhizae, of course. You don't really see many areas in the tropics of ectomycorrhizae. People think they are not very present there. They are not. This is something rare that maybe people don't want to invest a lot of time researching them, but I'm crazy about them. They are, and we know they have very, very different evolutionary histories, ecology, and if you saw that map of distribution of Quercus, it's really not that rare. It's just the number of species is very low, but maybe the area that they cover is not that small. So I did this translate review with two of my collaborators, Terry Henkel and Matt Smith, and we went and collected all the information that we knew about tropical ectomycorrhizal hosts. We found there were a lot of species that form ectomycorrhizal associations in the tropics. Found that Dipterocarpaceae, of course, is very important in Asia. Myrtaceae is very important in Oceania. Fabaceae is very important in Africa. And in the neotropics is very, very weird. But a lot of mountain species like Joglandaceae, Fagaceae, Betulaceae, and Polygonaceae and Nyctaginaceae are from the lowlands. And I'll also do another review trying to understand what were the important taxa in the tropics, in the fungi side. And we found that Rusula is so important, Inocybe, Tomentella, and these account for 60% of all the species richness that is identified in all tropical areas. So Rusula is my favorite genus. And it's believed it has an origin in the Afrotropics. We don't know yet, but it's extremely mega diverse in the tropics. So when I moved to Colombia 6 years ago, I basically had to start from scratch to understand the ecology of these tropical monominal forests. So, and this is one of my first students, Miguel Rendon, an undergraduate student that knows a lot about R, more than the average undergrad. And then we took, and here I want to totally second what Peter said about herbarium specimens. If they weren't by herbarium specimens, we wouldn't be able to really reconstruct this. So we took a lot, over 200,000 herbarium specimens from Colombia, and we assigned the mycorrhizal type, and also we included nitrogen fixers to see, and only in the Andean region of Colombia, to see their distribution along the altitudinal gradients and along soil environmental gradients. And of course, as expected, almost 82% of the species form arbuscular mycorrhizal associations, and only this tiny bit, 0.6%, form ectomycorrhizal associations. Those are the oak forest, the Nyctahyginaceae, and the Polygonaceae. Very little diversity. This is not abundance, only diversity. But when we went and looked at all those records, and we really found a pattern, like ectomycorrhizae, or no, the color is basically AM versus the rest. AM loves the lowlands and loves high nitrogen conditions. And along the elevational gradient, we found that Ericaceae really have its peak over 3,000 meters altitude. Nitrogen fixers decrease with altitude. For ectos, we really couldn't find a significant trend because there are very little records, and so on. So we try to model basically total soil, precipitation, temperature, and try to find where were the factors explaining the distribution. In another example of using fungarium specimens, we took the database of the biggest fungarium in Colombia, over 11,000 herbarium specimens, and tried to understand what was the distribution. But this was mostly across different forest types. So we divided between oak forests, mixed mountain forests, extra Amazonian lowlands, and Amazonic lowlands. And really, we found ectomycorrhizae fungi everywhere. But of course, the oak forests, they are more abundant. And Rusula, Amanita, and Cortinarius were the most abundant taxa in the oak lands, in the oak forest, and in the other ones, they were mostly litter decomposers dominating the community. And we did this very simple correlation network analysis, and we found that definitely the composition of mushrooms in oak were different, as expected. But this kind of gave us a better picture of what we were looking at, what were the distributional patterns. And actually, these quercus forests, even if they are not in least concern, they are in least concern category. They are affected, the mycorrhizal communities are affected by environmental conditions. Of course, we did this little study comparing urban versus natural quercus. We went in Bogota and nearby in a natural park called Chicaque, collected ectomycorrhizal root tips, and really found a striking difference. The natural forest is dominated by Lacterius and Rusula all over the place, while in Bogota we have Laccaria and Scleroderma, which I thought was amazing because I have done a study in Panama showing that in nitrogen addition plots, you have an increase of nitrophilic fungi, and those nitrophilic fungi were in the Laccaria. Also, Scleroderma has been shown to be associated to very disturbed habitats, so it made sense, and I thought it was really interesting. Of course, this is reflected in the NMDS. The cool thing about the Scleroderma, I went and collected the fruiting bodies, I sequenced it. I found it was a Scleroderma bovista, because I really don't know why these fungi are so different in between. They are not that far away. So are these trees being inoculated, or where are these getting all these fungi from? I found Scleroderma has a global distribution, so maybe they are being inoculated with some commercial inoculum. I don't know. It's a mystery so far. Okay, and then I'm going to jump into showing you tree propagation, the importance of this mycorrhizae for tree propagation, which, of course, I have to mention Colin's work and what he mentioned yesterday, this amazing paper showing that when seedlings are inoculated with live soils, from conspecifics, they grow better. For me, it was a concept that I have already thought about because these ectomycorrhizal fungi are, as I said, ectomycorrhizal inoculum islands. When you are only 6.6% of all the species, you know that they are limited by inoculum. So we did this cool experiment. This is not an ectomycorrhizal species, but it's in the Juglandaceae, and it's an arbuscular mycorrhizal clade, but I'm pretty sure they have very strange associations with fungi. So Mariana is another undergraduate student. She set up this experiment with this endangered species. We went out, very difficult terrain, dangerous area, with our mule, collected the soil inoculum, and actually did on-site the experiment in this area of Colombia. And she found really cool stuff. We planted seeds that came from 2 populations and inoculated only with inoculum from populations of one of the sites. And we found that, of course, seedlings that were inoculated with the native soil grew significantly higher than the ones that were not inoculated. Also, we found that we separated inoculum sources, each one from each one tree. We found that tree number 2 has the best inoculum. The other 2 weren't that great. So that inoculum 2 had different responses. So inoculum origin matters. Also, seed origin matters. We found that seedlings that were native from the site that the inoculum came from have a more positive response than the ones that were from a different. So Amoya were the local soils and Boyacá were the no-local soils. And Amoya responded more strongly positive to the local soil inoculum. So origin of the seeds matter. We also found a very concerning result that I have found with all my students is that the peat, sterile peat that we import probably from Canada is probably not as sterile as we think. This is not, we didn't sequence the sterile peat. Well, we sequenced the peat after we have germinated the seeds there. It has been exposed. Well, we found a lot of pathogens on that peat. Plant pathogens are plant pathogen, this. So this is native soil. These are native seedlings. And this is the sterile peat. And these are all the greenhouse-produced seedlings. Seems like this peat is leaving a legacy effect on the seedlings that were inoculated later with the native soil. I don't know what is going on. Really don't know. But I'm concerned that we could be inoculating these pathogens. And these pathogens come from Fusarium, Dactylonectria genus. So you see they are here in green. The native soil, native seedlings don't really have a lot of that, and they have more like a lot of saprotrophs. Okay, I'm going to show you another example, and I don't know how I'm doing on time, probably over time. Okay, another example, this is with an ectomycorrhizal plant. We were with Luisa, she's also an undergraduate student from Universidad de los Andes, and we were talking when we were planning on doing her research. We were talking to people in a botanical garden. They were doing restoration, and they were working with cocoloa. And I was like, oh, do you know cocoloa is ectomycorrhizal? Are you doing something about it? Nope. So we decided to do something about it. So this is in the dry forest, basically in the coastal area of Colombia. Sorry, I forgot to delete something. Anyways, she planted seeds directly on the inoculum or seeds in a sterile pit. And then those seedlings got transplanted either to native soil or commercial mix of soil. So we basically have four levels, two treatments. And what we found is that actually seedlings that were inoculated with the native soil grew better, especially the ones that were germinated directly in the inoculum and either they passed to commercial soil or native soil. It didn't really matter as much, but the initial inoculum that they were exposed to, it really matters. Sorry, the letters are a little confusing, but these are the ones that were grown in native inoculum, and these ones were the ones that were grown in a sterile pit initially. So we found that, and it was really interesting. We also found that, of course, the seedlings that were grown in native and transplanted to native have the highest ectomycorrhizal colonization. And, of course, there is a very concerning part 2 where we found a lot of seedlings that were grown in peat that had a lot of pathogens. Also, seedlings that were grown in peat and transplanted to commercial soil, they didn't even amplify. So we're missing some treatments here because they have no fungi in their root at all. So that was very interesting. Okay, that's another story. And I'm moving to the other story, and it's the other way around. So here is plants that needed inoculum to grow, but here I'm going to talk about fungi needed their host plant to survive as well. So how this host, tropical ectomycorrhizal host, can act as an umbrella species for the protection of their mycorrhizal species. So I'm going to show you a little bit of results that I have on this species. I show you that it's an endemic species. It's very, it's only present in Colombia. And these are my collaborators, Andrés Abella, professor at the Universidad Distrital, César Parra, which was working with us in the project, and this is another undergraduate student from Universidad de Antioquia. And we sampled in 2 populations in Santander in the north, and most of our sampling has been done in Huila. And these are very monodominant forests, high above ground biomass, very productive forests. I already said this. And we were able to establish permanent plots, measure, basically identify everything, measure every tree. They are super monodominant. They are up to like 96% Trigonovadanus trees in these forests, in these plots. So it's super dominant. We have collected a lot of genera, mostly Rusula. Rusula is incredibly diverse. These are results from Illumina sequencing from the soils. This light blue is Rusula. Is just overwhelmingly diverse. Here they are. These are other fungi that are very beautiful from that place. And we found only with 15 samples of Illumina that this host has over 200 ectomycorrhizal species. So if you have a very spatially limited population, very endangered forest, the most obvious thing is that these 200 species are also going to disappear. So I think we need to think about this species, these host species, as umbrella species for all the microbial biodiversity that lives on the soils. We also did this correlation analysis when we took the ectomycorrhizal fungal alpha diversity against the basal area of Trigonovalanus, and it's a positive correlation. So the more Trigonovalanus, the more species. But also, I took the other ectomycorrhizal species present in this forest, which are just 2 other species. They have a negative correlation, probably because these are all only Quercus and Alpharoa. Alpharoa is very a pioneer species. I don't know why this is happening, but it's an interesting trend. And we, of course, are finding tons of new species. Again, this is to make the argument that if the plant disappears and we don't even know who is there, we're going to lose them forever. And there are tons of new species. This is a collaboration I'm doing with Slavomir and Sik from Slovak Academy of Science. And of course, population also matters. This is a population in the north, and this is in the south. They host very different populations. So we need to protect several populations in several soil types and not only one population in one site, because we're going to lose probably half of the diversity. Future perspectives, well, actually, what is climate change doing to these very fragile environments, right? This is the cloud forest. We know that species are going uphill, right? In the tropical forest, they are going 10 times faster, it has been shown. So we are really in a rush to figure this out so we can protect. Why? Because, and this is both for the plants and the fungi and all the microbes, because we don't know, it's going to be a decoupling. Is the plant going to migrate without the microbes? Or are the microbes going to migrate without the plants? Or are they going to migrate together? We don't really know. We need to figure it out. And also the function of these fungi. So we have this new project I'm very excited about with a SPON and also with the JGI in RAI and Universidad del Rosario, where we are doing metatranscriptomics of the soils, genomics of the mushrooms themselves, and transcriptomics of the mushrooms themselves to actually understand what they are doing in these systems. And that will be it. Thank you so much.
So I'm just wondering, are there any existing conservation efforts to protect these cloud forests, or is there nothing in place at the government level?
At the ecosystem level in Colombia, no, not really. There are just national parks, things like that, but no.
But they are protected from logging some of these sites. So
in the national parks, but it's just...
There's lots everywhere else that can be harvested.
Yes, they are even cutting down the national parks there, wow. So they are very unprotected.
So it's really critical to
yeah, exactly. Jennifer.
So that is great.
Thank you.
So I have a comment and a question. So the comment is, I think you're right about scleroderma, because here in New England, if you look at the base of the street trees, you'll see scleroderma around the street trees in the city. And we do similar inoculation studies. We just started it, and when you walk in the ecological reserves, the forest reserves that are in the city, around now, actually, you'll see all these scleroderma pop up, and if you go out to the rural, they're not there. Yeah,
Cool,
so there's a
yeah.
lot of scleroderma in urban areas here in this part of the world too, which is really cool. I don't know what species it is, but yeah, it's very similar. So my
That's
question
interesting.
is, have you ever looked for ant colonization on your oaks?
Not in these oaks, but I did look at Oremonia. But yeah, I haven't had time to really get to the colonization part in these oaks.
Well,
But
I
yeah.
asked because it's interesting that migration study and
Mhm.
like that. But I also wonder if they're associated with ants because they can early in life. Right?
Yes.
In that very early stage. And maybe some of the other plants too that you're studying, like in addition to the ectolayer light, I wonder if the AM
great question. I definitely gonna look now, but I don't know. That's a great point. Yes, in Oremunia, which is another very similar cloud forest, but in Panama, I definitely saw colonization in seedlings and growing. When I transplanted them to the arbuscular dominated, they definitely show a little bit of colonization. Yes, that's a great point. Thank you so much. Yes, Michelle, then you.
Nice talk. I guess I'm curious if you're thinking about designing binoculars for like reforestation events. Like, do you just use multiple strains and just hope that the best one gets there? Like, what's the best way to even think about how to design a binocular?
Wow, that's a hard question because I don't know. Maybe Colin could be not responding this question. But what I've seen is that one tree could have many species at the same time. So designing an inoculum could be very, interesting, but I would just put as many species as possible, probably. But that's a great question. I don't really know. But could. Thank you. Yes.
Thank you so much. That was a wonderful talk.
Thank you.
A similar question in terms of like inoculum. You're talking about how local seeds are more responsible, so like are more responsive to soil inoculation, and the idea that like the symbiotic fungi are not like moving as fast as trees might need to, and things like that. Like, what are your thoughts? Are people talking about like moving soil, like moving local soil? Like, how do you? Obviously, we don't know the consequences of that either. So, are people thinking about that, or like, what are your thoughts around that?
I think for this is just local soil, and that's something that we really need to think very hard about it and experiment a lot before it happens. But it has the potential to really use it as a restoration tool because, it has been argued in the literature and calling a lot that this would help recover the underground, basically, biodiversity. So it's super important to think about that diversity, but we need more experiments, for sure. And in the tropics even, we're just starting. Yes.
What are the dominant modes of dispersal for the spores of the mushroom-forming fungi?
That's a great question. I don't really know. I think it's airborne, dispersal. For the most part, but I know, for example, for truffles, it's mammals as well. So it would depend of the group, but I think many of the mushroom-forming species are probably air dispersed. Hypothesis, totally a hypothesis.
Wonderful. Well,
thank you
so
Andrea. Much.
Okay, everybody. We're going to go ahead and get started in the interest of time. I already let us run a little bit late. I just wanted to welcome everybody back to the afternoon sessions, and just to reiterate what a phenomenal job all the people that put posters together did. I knew I was going to learn a bunch from the talks, and frankly, I'll be honest, I pitched the posters as being really great without having looked at them before, and it turns out they were really great. I learned so much over the month, so great job. Thank you all for putting that material together. It was wonderful. So with that, I'm going to welcome up Karina Berlingeri, who is a PhD candidate in the department, to introduce our first afternoon speaker.
I'm extremely pleased to introduce Dr. Jennifer Bachnagar, an associate professor of biology at Boston University. Jenny began her academic career at BU as an undergraduate before doing her PhD at UC Irvine, and then postdoctoral work at the University of Minnesota and Stanford. She then came back to BU to join the faculty, where she runs an incredibly productive lab and has won numerous distinctions for her teaching and mentoring. This stellar mentoring track record includes serving as a postdoctoral advisor for last night's speaker, Colin Averill. Jenny has also received a highly competitive Bullard Fellowship at Harvard Forest to work on the shifting plant-fungal relationships with historic land use. The focus of the Bachnagar Lab is understanding how microbial communities change in response to human activity and what that means for microbial function in an ecosystem. I think Jenny is best known for her lab's expertise in molecular techniques that allows them to quantify microbial community composition changes and link those changes to ecosystem function. Fittingly, today Jenny will tell us about meta-omics insights into ectomycorrhizal effects on soil carbon cycling. Please join me in welcoming Jenny.
Thanks, Karina. And thanks to you all for coming back after lunch. Good job. You made it here. And thanks, Ben, for putting this on. This is so much fun. This is a fun day. So I am going to talk about what's in this title, but it's a really boring title. So I propose a change to the extraordinary capabilities of ectomycorrhizal fungi. That's one of the main groups that my lab focuses on. Oh, and how we're changing them. So we've heard a lot about these organisms today and yesterday. They are diverse, the ectomycorrhizal fungi. They are functional. They can provide up to 80% of plant nitrogen, we think. And because of how they do this, these fungi have historically been considered scavengers for non-carbon nutrients, nitrogen, phosphorus, sulfur, cationions, things like that. But we know that ectomycorrhizal fungi can act as decomposers themselves. This is a big area of research, for better or worse, for me and my lab, because if they do this in reality when they're attached to a plant, it means that the release of carbon from soil through respiration, decomposition respiration, is not just regulated by the activity and the composition of the microbiome. It's also regulated by the physiology and functioning of the plants that the microbes are attached to. Who cares? Well, this is really important, especially for modeling, recapitulating how carbon and nitrogen cycles through ecosystems in our conceptual and predictive models, and Eddie's going to talk about that next. But there's this other thing. So global changes are happening in fungal habitat, and the global change that I'm most interested in these days is in urbanization. So I'm going to talk a little bit about that at the end of this talk. So urbanization is interesting to me because it's the fastest growing type of land use change on Earth. It's the fastest growing biome on Earth, urban ecosystems. By 2050, 90% of the US population will live in cities, and urban areas will subsume 20% of US forest land. That's about the size of Pennsylvania, and that's probably an underestimate. So I'm going to talk about two AMF stories today. The first is answering the question, how do ectomycorrhizal fungi influence carbon cycling on a very fundamental, tedious molecular level? What we know about it, what we don't. And then I'm going to talk about the impacts of urbanization on that process. So let's start with number one. How do ectomycorrhizal fungi influence carbon cycling? Well, we know a lot about this from the plant perspective, the big plant, little plant experiments that Colin talked about yesterday. Below ground is a little trickier to sort out. So there's this hypothesis that if ectomycorrhizal fungi act as decomposers, they're going to decay especially nitrogen-containing organic molecules in soil. If they do that, they might actually compete with free-living saprotrophic microorganisms for nitrogen. They might suppress the growth and activity of those saprotrophs, and that would lead to a suppression of soil carbon loss. And this is a popular and kind of famous hypothesis called the Gadgil effect that was published in the 70s. But in many ways, it has not been very well tested, especially in the molecular level. So Colin, when he was a postdoc in the lab, wanted to test this, and he thought that if you add nitrogen fertilizer, it should shut down this whole thing. There should be no competition for N, everyone's happy, no suppression, soil carbon loss. So to test it, Colin and a whole bunch of other people set up some experiments in the greenhouse. And the experiments I'm going to talk about today included a model plant ectomycorrhizal system, pine seedlings and their ectomycorrhizal fungi in the genus Swillis. And we grew them in these tiny ecosystem boxes with a little respiration chamber. Oh, there's a pointer. Oh my gosh. The top. Yeah, okay. So why? Oh, so what we did is we grew the pines, but we grew them in an agriculture soil from a maize field, right? As you do in ecosystem ecology. No, wait. So the reason is because you have this differential in the deltas 13C in the carbon from the C3 plant and the C4 soil. And it's big enough that when you measure the isotopic composition of your CO2, you can partition the respiration from the decay of soil carbon versus respiration from the plant. Super great if we want to figure out ectomycorrhizal effects on soil carbon decay. So in this system, we add the ectomycorrhizal fungus that the pine associates with, or we take it away. We can add all sorts of levels of nitrogen, and we measure all sorts of biogeochemistry and molecular metrics of fungal activity. And so what we expected was that when ectomycorrhizal fungi are there, they are going to suppress soil carbon cycling if they suppress the activity of saprotrophs. So in red, you have samples with the fungus, the ectomycorrhizal fungus, and it's suppressing soil carbon-derived respiration, and that effect goes away as you increase soil nitrogen. However, this is not what we find. In this experiment, the presence of ectomycorrhizal fungi increased soil carbon respiration. Why? When we look at the metatranscriptome, we see that the presence of ectomycorrhizal fungi, which is here in red, it actually tends to suppress the secretion of proteins that break down soil carbon molecules. You can see what some of those proteins are here. But the ectomycorrhizal fungi themselves are dumping these out at extraordinarily high levels. Okay? So be very clear about this. Ectomycorrhizal fungi are secreting proteins outside of their cell that break down soil carbon molecules. Okay. Yeah, okay. However, when we look at sugar uptake transporters, it turns out that while ectos are shutting down soil carbon degrading enzymes by saprotrophs, they're ticking up the sugar transporters expressed by saprotrophs, both the saprotrophic fungi and the saprotrophic bacteria. Okay, so the presence of ectos is suppressing actual decomposition of soil carbon by saprotrophs. They're decomposing soil carbon themselves, and they're increasing the uptake of that carbon by the saprotrophs. When we put all this together, it's both the extracellular carbon decomposition enzymes and the extracellular sugar transporters that together kind of best explain these soil carbon-derived respiration rates. Interestingly, while these Swillis ectomycorrhizal fungi suppress carbon decomposition by saps, they actually increase nitrogen decomposition to some extent. So they're secreting, these are mostly proteases, amine oxidases, some chitinases, and yeah, that's mostly it here. So it's quite a diversity, actually, of enzymes that the saps are putting outside their cells to access soil nitrogen. But going back to the original idea of what ectomycorrhizal fungi do to saprotrophs and what the impact on carbon is, I think in this case, there's not great evidence that ectomycorrhizal fungi are suppressing the activity and growth of saprotrophs. Instead, it looks like the ectomycorrhizal fungi themselves are driving soil carbon decomposition. They seem to be feeding, essentially, the saprotrophic bacteria and fungi, and potentially even priming the decomposition of soil nitrogen. And this together is leading to increased soil carbon loss when the ectos are present. So I remember when Colin first talked about this project at ESA some years ago, I was thinking about it, like, what is going on? Why are ectomycorrhizal fungi not suppressing the other organisms in this system like you would expect? And it took me a few minutes to figure it out. And I wonder if anybody has an idea about why that would be. What do you think? No, don't answer. You don't, but yeah, what do you think? Eddie knows the answer. But
they're not co-evolved with those communities.
Okay, so maybe there's a coevolution issue. So these saprotrophs have never seen an ectomycorrhizal fungus before, never seen a pine seedling. Okay, so why would that matter to carbon cycling? Yeah.
Are your soil nitrogen mostly found in the MAM pool in these agricultural soils as opposed to particulates that you would find in EM forests?
Yes, so that's a good point. So the point is the nitrogen, organic nitrogen, might be mostly found in the MAM pool as opposed to the particulate pool. Yes. So it turns out in this system, I haven't looked at nitrogen in the POM and MAM, but when you add the ectomycorrhizal fungi to the system, POM goes up, as it normally does in an ectosystem, and MAM goes down. And the only way I can explain that right now is that I think that ectomycorrhizal fungi, with a little bit of help from saprotrophs, are breaking it down. So they're accessing it. So there's another reason that I think this happened, and it's related to the agricultural soil. We grew this in soil from a cornfield from Indiana, middle of Illinois, Champaign. Best corn soil, except, how many people have been to a cornfield? Everybody, oh my gosh. I'm so impressed. Okay, so we're here. How many people have been there when the corn is harvested? What does it look like? What's it look like? Brown, stubble. It looks like this. When corn is harvested through conventional agriculture, this happens, and we just take all that plant material away. It's gone. Like, they put it in little balls, and they send it off to do something else in the world. So I actually think agricultural soil microbiomes are starved of carbon. We didn't induce nitrogen limitation in this experiment because they were so severely limited by carbon. So when you put a delicious plant in there, of course they're going to go totally nuts. They're going to stop trying to decompose whatever bits of carbon are left there. They're going to eat the carbon provided by the roots and the mycorrhizae and be quite happy. So when I brought this up to Colin, his response was, well, let's dose up the carbon. So that's exactly what we did. If you increase soil carbon, maybe you induce nitrogen limitation, and then maybe you get this competition effect. So to do that, we redid the entire experiment over the course of a whole nother year, and we added a whole bunch of corn stover in to get a bunch of carbon in there. And when we do that, we see what we expect, which is that when you add ectomycorrhizal fungi to the system, it starts to suppress the decomposition of soil carbon. when you look at the metaproteome and the metatranscriptome in these data sets or in these samples, we see that the presence of the ectomycorrhizal fungi tends to decrease, again, the secreted soil seed decomposition genes, primarily by bacteria, actually. The saprotrophic bacteria respond pretty strongly here. But it turns out that the presence of the ectomycorrhizal fungi generally are decreasing the metabolism of the saprotrophs. And you can see that here. So this is actually the bacterial peptides from the saprotrophs that code for a bunch of enzymes in the central carbon metabolic processes like glycolysis and stuff. And that's the best predictor of soil respiration rates in this experiment. However, when you look at nitrogen cycling again, this is a place where the presence of ectomycorrhizal fungi tends to tick up the activity of the saprotrophs. They tend to increase amino acid uptake transporters quite a bit, and you'll notice that that's suppressed under high nitrogen. This is pretty common in this experiment. But it also increases the production of antimicrobials. So these are multi-drug efflux pumps in the bacteria, but also the fungi, saprotrophs, tend to tick up. And actually in the ectomycorrhizal fungi too. And so what I think this means is that there's a chance we actually did induce some kind of competition for nitrogen, and there's a lot of antagonism in the system. So for the first part, how do ectomycorrhizal fungi influence carbon cycling? Well, I think one thing we found here is that ectomycorrhizal fungi can decompose soil carbon all by themselves with their own enzyme systems. And they can change the amount of soil carbon decomposed by saprotrophs depending on the amount of carbon and nitrogen in that system. Okay. I'm not sure how much time I have left. What do we got? A little bit? 10 minutes? Okay. So I want to move on and talk about number 2. What are the impacts of urbanization? So, oh wait, before I do that, I'm going to introduce Eddie and Eddie's work. So part of us going through all the trouble to figure out the fluxes of carbon in these EMF, non-EMF systems and the ways that the ectos and the saps are using their carbon is to build this into some ecosystem models to get better sense, especially of carbon allocation by the plant, but also of the fungi and the bacteria into what they're doing down there. And he's going to talk about that next. But before that, I want to talk about urbanization. So urbanization is this global phenomenon. It has a really interesting mix of global change stressors that are relevant to plants and the microorganisms that colonize them. One of the interesting consequences of urbanization that you don't get with other types of global change is fragmentation of habitat. And so what we wanted to do in our study of urbanization is look at the interactive and potential compounding effects of urbanization and fragmentation on forest fungi. And so to do this, I joined a project with Pam Templer and Lucy Hutira at BU called Urban New England Project, which consists of oak-dominated forest plots from Boston all the way out to Harvard Forest. And at each of these plots, we have this transect from the forest edge 90 meters into the forest interior. So we have a lot of data from this gradient study on pollution loads, above ground plant responses to urbanization and fragmentation, and we came in to do the microbes. And this work was driven by Chikai Tatsumi, who's here today. She's a postdoc in my lab. And what Chikai did is she started by sequencing the DNA in the soil, and what she found is that the ectomycorrhizal fungi do not decline with urbanization. They decline with fragmentation. They go down at the forest edge, and you can see that here. So this is the fraction of the fungal community in soil that is ectomycorrhizal for urban forests in red and rural forests in black at different distances from the forest edge. And you can see, right, they go way down to the forest edge. This happens despite the fact that you have the same above ground productivity of ectomycorrhizal trees at the edge. You have the same root biomass at the edge. Yet we have this pretty serious decline. The other thing I want to point out here is that urban forests have a lot of ectomycorrhizal fungi in them. However, when we look at the roots, and this was work done by Emerson Conrad Rooney, when we look at the roots and you look at the colonization of the roots, in the urban forest, root colonization is much, much lower. So this is a lesson for everybody who is interested in molecular stuff. You can measure DNA, you can measure all the DNA you want, but it doesn't mean that those microorganisms are active and doing the thing you think they're doing. They most certainly are not in this system. Yeah, yeah. Okay, but DNA is somewhat useful sometimes, right? Something is going on here with DNA. Okay, now, and this is another thing we can do with DNA. So my PhD student, Katie Atherton, constructed networks just based on the DNA, ITS amplicon sequences of, and the 16S, together bacteria and fungi in urban and rural forests. And there's a lot to talk about here, but one interesting thing is that the microbiomes in the soil of urban forests are more connected. The taxa are more connected to one another. You can see that in the edge density and transitivity plots. But when you look at just the ectomycorrhizal fungi, they're basically disconnected from everything else in urban systems, despite the fact that they are very, very abundant. So, what's going on? Well, we thought that maybe they're dead, right? They're not colonizing plants. Maybe they're just laying there dead or dormant. No, no. So this is new data from Chikai. This is the metatranscriptome of all the fungi in soil. Oh no, I take it back, of the ectomycorrhizal fungi in soil. The urban edge, urban interior, rural edge, and rural interior forests. Take a look at the urban interior. It's all red, except for nitrification. They don't do that. Everything else they are doing, and I really think this is evidence that in the extremely stressful environment of urban ecosystems, ectomycorrhizal fungi, they just don't follow the rules. They're doing something totally different, and I'm wondering if they're acting even more as decomposers in disguise. So that's something that we're looking more into now. The last thing, the thing I want to leave you with, is that as ectomycorrhizal fungi decline, in this system and many others, the pathogens increase. And this happens for both the plant pathogens and the animal pathogens. And in fact, we're seeing this in city trees as well. This is in the soil and the roots, as well as in the leaves. And this is work that's being driven by my PhD student, Katie, who I mentioned before. She's looking at street trees all over Boston and finding that in the leaves of these street trees, we do have higher pathogen loads, particularly something called sooty molds, which is just like a group of potential plant pathogens. But also we're getting quite a few more foliar endophytes. So what I think this means is that urbanization has a generally sort of negative impact on ectomycorrhizal activity. Certainly forest fragmentation does, and to be honest, I can't quite figure out why. I can't quite figure out what the environmental condition is at the edge that causes this massive decline of ectomycorrhizal fungi, except for just physical disturbance. I actually think that might be the most important thing, but that's TBD. But it's certainly, urbanization in general isolates them in their activity, so they're not very well connected to the rest of the microbiome. And urbanization also appears to select for alternative tree-associating fungi, such that there is a chance that trees in urban systems are operating by a totally different rule of life in how they interact with fungi. So that's it for me. Thanks to everybody here today. Thanks to my collaborators and my students, and I'm very happy to take questions. Okay.
Do you have, in your urban stuff, is there more invasive plants? Is there like, is there a pile of garlic mustard or knotweed or some other crazy?
So we've done understory, and the person who knows that is Corinne, my PhD student Corinne. Do you remember the understory of the urban forest, if it's radically different than the understory of the rural forest in your sites?
It's not radically different, but the understory changes a lot at the first edge. So at the first edge, you see like a lot more ferns, a lot more taller under like grasses, a lot more literally just like bare soil. So I think the edge to interior is stronger than the urban rural.
Yeah.
Okay, so it's not like there's a pile of garlic mustard sitting there.
No, not in these sites. There might be others, but it does bring up an interesting point that maybe the roots that we were detecting at the edge are not tree roots. I mean, I think they tried to make sure that they were woody, but I think we have so much other stuff there that maybe we're just getting more non-ectomycorrhizal roots at the edge. Yeah.
That was sort of along the lines of my question is because the edge to interior graph is the percent of the community that is EMF, right? And so how much do you think that is attributed to a drop in EMF versus a bump up in other stuff?
So we've been through the ringer on that, and we've tried to figure out what is real and what is not real. And I actually think, because no matter how we normalize it, no matter what we do, I think this is a drop in ectos. I'm not sure the extent of that might be up for debate because how you normalize it affects the extent of the drop. But my sense of this, and I can't confirm it because I don't know for sure, my sense is that when the ectos are there, the saps drop rather than the other way around. And that's from like experiments when you kind of inoculate and too. So I think that's what's happening, but I'm not 100% sure.
That's super interesting. Thank you so much. For the urban part, I'm super curious about the transcripts. Do you correlate that, for example, with a change in species composition or soil?
Yes. So these are gradient studies. So there's definitely a change in composition just because of space, right? That's the best predictor of composition. It's just distance between sample points. We haven't backed that out of the metatranscriptome, but we have backed that out of the DNA data, and there's still a very strong urbanization effect. But we do have to do that here too. In fact, I think the ectomycorrhizal DNA that's like annotated as ectomycorrhizal is a very strong correlation with the ectomycorrhizal RNA. So I think we'll probably see similar things. I wasn't sure that we would, but it's a little encouraging, yeah. But we'll look for that too, like what the composition of these breeds are, like which taxa are doing the jobs. Yeah, that will be very interesting. Yeah, Peter.
You've been pretty careful with your words about ectomycorrhizal fungi as decomposers.
Have I?
Do you get the sense from your isotope data, if you look in the hyphae, are they picking up any of that corn seed and putting it in their bodies and acting like saprotropes as well?
I would love to know. I don't know if we have the differential to do that, but maybe. Yeah, duh. We should totally do that. It's a really good idea. Yeah, and I think in some of the other work that my students will do, that's one of the ideas is to put labeled litter down and look at it in the root and, try to do our best to make sure that it's going inside the fungus and not somewhere else. It's really hard because these fungi are associating so closely with the bacteria. It'll be a tricky business, but yeah, I would love to. It's a good suggestion. Okay, thanks everybody.
Hi, everyone. My name's Elena Bisson. I'm a research assistant in Dr. Ben Taylor's lab. And it is my great pleasure to introduce Dr. Eddie Brostek, Associate Professor in the Department of Biology at West Virginia University. Eddie did his undergrad and graduate work right in our backyard at Boston University, and then went on to do a postdoc with Rich Phillips at Indiana University before joining the faculty at West Virginia. He has won several awards, from the Northern States Research Cooperative to the Ecological Society of America, to more recently serving as a West Virginia University Bridge Faculty Policy Fellow, where he worked on connecting policymakers with the science behind carbon markets and natural climate solutions. Work in the Brostek Lab spans topics of empirical biogeochemistry, climate change, sustainable bioenergy production, and plant microbial interactions. But where I think Eddie really stands out is in his incorporation of the mechanisms that drive complex ecosystem processes into models that help us predict the future of terrestrial ecosystems under global change. Today, it sounds like Eddie will talk to us about how challenging those efforts can be at times, as he tells us about the long and winding roads towards integrating root microbe interactions into ecosystem models. Please help me in welcoming Dr. Eddie Brostek.
Thank you. Thank you for that wonderful introduction, and it's been awesome being here. It's been a blast being here over the last day and a half. Got to see the Arboretum, got to see some bonsais, got to smell some lilacs, and got to reconnect with some Boston University friends. Ben inadvertently made a Boston University Symposium halfway. So go Terriers, even though we're not really that patriotic or school pride. All right, so I just want to start off with, these are just some cool, this was a root exhibit that they had at the National Botanical Garden right in DC, so I always like to take a cool picture for talk slides. And this was a really cool tree that I saw in Greenville, South Carolina, so I just want to throw that in there too. All right, so first I just want to start off with some acknowledgments. So I just want to acknowledge the folks at West Virginia University that I work with, including Ember Morrissey, Karen Allen, who was a postdoc in the lab, who's now in New Zealand at Manaki Whenua Land Care Research. Jeff Walkup, who's a PhD student who worked with Ember, is now a postdoc with Steve Allison at UC Irvine. Steph Juice, who's a current postdoc. And then my all-important office mate back in the day when I was a postdoc, Ben Solman, who's now a research scientist at Oak Ridge. And then also collaborator Renato Bergire at JPL. And then these are the folks who kind of made me who I am. So this combination of models and experiments. So Rich Phillips and Josh Fisher are two folks I worked with as a postdoc at Indiana. All right, so Elena did a great job introducing me, but who am I as a scientist? I'm someone who's really interested in how processes that operate at the level of roots, mycorrhizae, and soil microbes in the rhizosphere impact ecosystem responses to global change. And so what we do is in my lab is we make measurements of things that are happening here, or we also do experiments, and we try to use that data to improve our ecosystem models so that we can make predictions of how our ecosystems will respond to future anthropogenic change in the future, and we're really focusing on soil carbon and nitrogen cycling. All right, and so where we're going today is, what I want to do is try to address this grand challenge. And this grand challenge is that empirical advances in our understanding of root microbial interactions have consistently outpaced what we've been doing with the models. So we're always playing catch up. And we have known for a long time that the rhizosphere and mycorrhizae are really important. So I really like this paper from Burns back in 1985, where he recognized that the rhizosphere is really important, but he also did stuff that I published like 30 years later, just looking at enzyme activities being stimulated in the rhizosphere versus the bulk soil. In addition, we've known that mycorrhizae are really important. This seminal paper by Sir David Reed and Perez Moreno kind of put ectomycorrhizae, arbuscular mycorrhizae, and aerocord mycorrhizae on the map and really showed that they're important for ecosystem carbon and nitrogen cycling. And we've even continued to refine these ideas over the past decade. So we've had work looking at how rhizosphere processes can both inhibit and promote rhizosphere microbial activity. I helped Rich develop the main framework. This is really his baby. He was like, get me some modeling stuff in there, so I wrote that stuff for him. We also have the work by Collins Group looking at how
Ectomycorrhizal fungi can enhance plant growth. We recapitulated what Burns did and showed that rhizospheres are more active than bulk soils. And then we even had this really great new special issue in New Phytologist showing that different mycorrhizal fungi differ in their enzyme capacities. And we were starting to get these new conceptual models of how arbuscular mycorrhizal fungi impact soil carbon cycling. And when you look at the models, we basically have none of this. So if you look at a large ecosystem or Earth system model, they're mostly based on this old framework in Biome BGC. And if you look at them, roots are simply a carbon and nitrogen pool that lacks function. So it's just a place where you can stick carbon and you can stick nitrogen. And one of the ways they do have function is that they impact water uptake. So if the root is there at depth and there's water at depth, you can have one root in the model, it'll take the water out. That's the one place in these large ecosystem and Earth system models where roots have function. And so what we're going to do today is we're going to journey through some important model advances that have helped bridge this gap. This list is not exhaustive. I apologize if you have a favorite model and it's not in here. I'm also going to talk about our efforts, kind of my journey from being an empiricist to becoming a modeler, and then also highlight some challenges and opportunities moving forward. All right, so let's start with this journey. All right, so even going back to 1991, right? To me, that's a long time ago. It's 30 years. The Phillies still sucked. The Red Sox hadn't won the World Series yet. But people were looking at predicting optimal carbon allocation to mycorrhizae. So this is a cool model by Fitter, where he looked at what the optimal colonization rate would be, where you would maximize or minimize the carbon going out and maximize the carbon going in. So you'd maximize plant growth, and you maximize that carbon cost of nitrogen acquisition through this plant. We even had microbial explicit models, and so we looked at how roots could impact microbial activity. And this is a really cool, I dug this up last week. It's a model by Blagodatsky and Richter, and they actually had a microbial explicit model in here where they had enzyme kinetics, they had microbial physiology, and they were even doing really cool experiments like here where they simulated root exudation. So in the top, you have a model that has no root exudation, and I want you to focus on this CO2 line. You see that it's pretty flat, it's not going up a lot. But in this one, they added exudates, and you can see that CO2 jumps up and goes really crazy. You get a lot of decomposition. And so we're even modeling these things back in the, where are we at here, 1998. Pretty good. And then most of the folks who model, this is one of Colin's favorite papers in the world. He loves it. So this Schimmel-Weintraub model really kind of moved us, even though it was a theoretical model, it actually led to a lot of applications in larger ecosystem and Earth system models. And so this is a really important model framework, and Josh will say it's a toy model, but what he did is he moved the field from first order decay to microbial explicit function in the models. So in your old school model, you have a soil organic matter pool, you have a decay rate, you multiply the two together, and you get decomposition. In this model, the microbes actually have function. They make enzymes, they have carbon use efficiency, they take up carbon and nitrogen. So you have that explicit microbial function. He was able to simulate the carbon and nitrogen limitation of microbial function. So it wasn't just carbon, it was both carbon and nitrogen. And again, at some level, this forms the basis of all microbial explicit models. We all use this, we all kind of tweak it a little bit, we all have our little things that we like to do, but we're all really based off of this model. And Josh and Mike were able to also simulate the effects of root exudates on microbial activity. And so here you're looking at microbial biomass over days. They add a carbon pulse here at day 25, and you can see you get a big pulse in respiration in the model, but you get the sustained increase in decomposition that lasts. So they're able to simulate this priming effect. And so we were doing a better job at free-living microbes, but what about mycorrhizae? How are we getting mycorrhizae into the models? And so this was a paper by Kate Orwin and also Ian Dickey. I had the opportunity to meet Ian a couple weeks ago. He peppered me with a lot of difficult questions. And so this model, the MyScan model, the real big advance here is that they actually put mycorrhizal fungi in the model, and the mycorrhizal fungi were able to take up organic sources of nitrogen. And they looked at the implications of that for soil carbon cycling. And so what they found is that when you had organic nitrogen uptake by the mycorrhizae, it increased soil carbon. And so here you're looking at, there it is. looking at soil carbon change across a number of different fertilities and litter quality. But you can see all across all these simulations that you got more soil carbon when ectomycorrhizal fungi were taking up organic nitrogen. One thing I will point out with this model, it doesn't have microbial explicit function. It's based off of century dynamics with that decay rate function, or first order decay. And we even got into this place where we're also looking at evolutionary stable strategies, one of Colin's favorite things, again, where we're thinking about, this is work done by Oscar Franklin, where he took kind of an economic perspective and was able to take these different strategies and show that in his model that mycorrhizae were able to drive nutrient limitation. And this is a really cool model, but we were still in theory land. And so this is sort of where my journey, when I kind of jump into the journey, and I'm not saying I pushed this whole field forward, I'm just telling you about what I did. And so here's some of the, here's where I start. And so this is a really old picture. This is actually my LinkedIn photo. It's kind of embarrassing. I haven't updated it since 2010 when I went to the Harvard Forest for a graduate student retreat. This is on the fire tower there. So this is me. I was Mr. Dr. Green postdoc. I was coming from Boston University, and I was working with Josh, who was Dr. Model at JPL, who's now at Chapman, and Rich, who was Dr. Roots and mycorrhizae. And we were getting money from the DOE, so our goal was to make me Dr. Modex, or Model Experiment Fusion. And one of the first things I did as a postdoc is Josh had worked and developed this model called the fixation and uptake of nitrogen. And what it was designed to do was to calculate a carbon cost of nitrogen acquisition and do a better job at representing nitrogen limitation in Earth system models. And what I did is I added in arbuscular mycorrhizal and ectomycorrhizal uptake. And so in the model, what you have is you have net primary production, and some of that you can spend on growth, and the rest you're going to optimally allocate to take up nitrogen through a variety of different pathways, whether that's retranslocation from leaves, biological nitrogen fixation from the atmosphere, or arbuscular mycorrhizal, ectomycorrhizal, and non-mycorrhizal uptake from the soil. And so what we did is this model is parameterized to have different cost functions and different optimalities. And so it doesn't just say, okay, if this is the cheapest cost, we're just going to fix everything. It tries to predict the optimal suite of best nutrient acquisition strategies across all of these. And so these are just some curves illustrating how the model works. So here you're looking at the cost of nitrogen acquisition for arbuscular mycorrhizal uptake in green, ectomycorrhizal uptake in blue, and non-mycorrhizal uptake in red. And you have constant root biomass and then soil nitrogen availability. And you can see that ectos were more advantageous or had a lower carbon cost in the model at low soil nitrogen. And then the model predicts that as soil nitrogen increases, AM becomes more advantageous. And similarly, when we keep soil nitrogen constant, we see that at low root biomass, the model predicts that AM is going to be more advantageous because AM are thought to be better at scavenging nitrogen. And so what I did then is, after I developed the model, we then validated it and tested it across a number of different sites. And so things that the model can do is it can predict, you can see here, it predicts the different suite of nitrogen acquisition across a number of different phase sites and some other long-term research sites. And the other thing that was really important is we had Fun 1.0, we had another model, and of course we had to add another number. But anyway, this is the Fun model now. And so we found that anywhere around from 4 to 15% of NPP was being spent on nitrogen acquisition. And so we validated the model at 6 sites, and then we said, let's make it global, all in the Eastern Temperate Forest. And so this was work that I did in collaboration with Mingji Shi, who's at PNNL. Her and I spent a lot of time at my dining room table in Indiana for a couple weeks figuring this out. So we took Fun and put it in the community land model, which is written in Fortran, which is a nightmare. But we were able to do really awesome things like do these global scale predictions of how much gross primary production is spent on nitrogen uptake, and look at how that impacted carbon cycle climate feedbacks moving forward. We even added in phosphorus. So this was work done by my former postdoc, Caro, who's in New Zealand. And so Michelle did a really nice job teeing this one up because she talked about, so in the model we have a nitrogen cost for phosphatase enzyme activity production. We also include organic acids and other things so there's a nitrogen cost to phosphorus uptake. And so you have a carbon cost of getting nitrogen and a carbon cost of phosphorus uptake, so it kind of compounds and interacts through the model. And so we tested this across a number of tropical and temperate forest sites. So the tropical sites were dry temperate forest sites from work done by Jennifer Powers in Costa Rica. And then the temperate forest sites were our mycorrhizal gradient in Indiana. And you can see the great thing about models is you can run experiments that you can't run in real life. And so we were able to do this experiment where we changed nitrogen, phosphorus, and nitrogen phosphorus availability. And we found across almost all of these simulations that these dry tropical forests, their carbon cost of getting nutrients was more sensitive to the removal of this nutrient limitation than the temperate forests. All right, and then we can then we add the phosphorus in, and then we go global again. And you can see that we get these, Renato made this really beautiful, I mean, that's a great conceptual model. I'm not going to walk through it, but just look at how beautiful that is. I mean, he even has the dollar signs. Like, I think I want to put dollar signs in a paper. And so the awesome thing that Renato is able to do is then he's able to run these global scale simulations using ELM with fun nitrogen and phosphorus in it and see how having these limitations or the carbon cost of nitrogen and phosphorus acquisition influenced GPP in the historical record. So here you're looking at global, sorry, not GPP, MPP, global MPP over time. Here's with no limitation, here's nitrogen limitation, and we even downregulate it even more with nitrogen and phosphorus limitation. All right, so I'm showing lots of bad pictures of myself, so I had to go back and find these. So this is a picture of me when I was a postdoc. My one friend at West Virginia has this on his office wall to make fun of me because I look really goofy in it. And so, but it was really serendipitous that at the time I was at Indiana, Ben also started a postdoc with Kim Novick, and we were stuck in this same office in this Hogwarts-like building at IU. And we only had like one window, and it wasn't near any of our offices. But in the time when we weren't walking to the coffee shop in the art museum, we decided that, hey, we both have these two models, let's stick them together. And so I was working on the fun model at the time, and then Ben developed the corpse model. And so this is the carbon organism, like a rhizosphere and protection in the soil and environment. It's a terrible name. The reviewer, one of the reviewers asked him if he wanted to change it. He said no, he really liked it. And it's stuck around forever. But the great thing about this model is really awesome because it includes a rhizosphere soil compartment, a bulk soil compartment. It has explicit microbes. And then the other thing that's really awesome is it simulates the formation of mineral-associated organic matter. So you have protected soil organic matter and unprotected soil organic matter and particulate organic matter. And it simulates that by saying that microbial necromass is the predominant form, or when you produce more of that, you get more mineral-associated organic matter. And so then we worked together to put these couple these two models, which is not trivial. People will be like, just couple your model together. It takes months, because when you start writing in code, you make bugs. But then you also come up with one of the more unfortunate acronyms that we have. But the really great thing about FunCorps is that what we have is a coupled plant microbial interactions model where Fun predicts how much carbon the plant is sending to rhizosphere microbes and mycorrhizae. That carbon then flows into corpse where it stimulates microbial decomposition, has impacts on soil carbon cycling, and then leads to the return of the nitrogen back to the plant. And so we then tested this across a gradient of 45 forested plots in Indiana that varied in mycorrhizal association, going from no ectomyc, so 100% AM to 100% ECM. And here you're looking at measurements of the rhizosphere carbon allocation or exudation to the rhizosphere as across that gradient. And then you can see that the model does a pretty good job of predicting that. It's not perfect. It's not getting the variability, but it's capturing kind of the mean response. And then we're also able to show that here you're looking at what are the impacts on soil carbon for every additional unit of nitrogen mineralization that you get through rhizosphere priming. And the model predicts that this is greater when you have all ECM trees than when you have all AM trees, suggesting that these are priming soil, at least in the model land, priming soil carbon more than the AM. And we have continued to refine this model over the years. And so we've applied it to bioenergy systems. So my postdoc, Steph Juice, has taken this into bioenergy. And I think one of the really cool things that she did with this model is she simulated tillage kind of mechanistically. So instead of in traditional models, when you have a tilling event, you just ramp up decomposition for some set period of time. So you bump up that K. We actually have protected soil organic matter becoming unprotected, kind of reflecting that breakup of soil organic matter protection with tillage. And then Ben also decided he was going to go global. And so this paper is really great. What he did is he took the Fund Corps Foundation, but then he tweaked it a little bit so it had more of a return on investment framework. And he didn't call them AM and ECM, but he had nitrogen fixers, nitrogen miners, and nitrogen scavengers in the model. And then he ran it globally. Of course, we've got to run it globally. We did validate it at Duke and Oak Ridge, so it wasn't like we didn't do any validation. But the really important thing that Ben was able to show here is that here you're looking at simulations of how the carbon storage changed across the globe when we elevated CO2 by 100 ppm. Here's the carbon stocks, here's time. I just want you to focus on the green, or sorry, not the green. The green is no limitation. You can see that carbon stocks just go up. But if you look at the blue and the orange, the blue is when we allowed plants to shift their nitrogen acquisition strategies or ecosystems to do that. The orange is when we let it stay static. And you can see that when you have dynamic shifts in nitrogen acquisition strategies, you get a sustained increase in soil carbon stocks or soil carbon accumulation in the ecosystems. Here you can see that this is predominantly driven by greater nitrogen uptake. Makes sense. All right, so what are some of the challenges and opportunities moving forward? So one of the things I think is we still are a long way from explicitly representing differences in mycorrhizal physiology and traits. So Jenny was at this conference, and I don't know if anybody else in the room was, but we had this great meeting in Amsterdam where we got wined and dined, and we were going to get mycorrhizae into the models. Well, we got a book chapter. Please read this book chapter. Take a picture. There's a lot of work in here. But one of the things is we made this really nice conceptual model, and one of the things that came out of that meeting is we still lack fundamental understanding of how much carbon plants are sending to mycorrhizae, what the mycorrhizae are doing with that carbon, what's going to growth, metabolism versus resource capture, and then how is that feeding back on the microbes in the rhizosphere. And so what happens in our models is we tune all those parameters so we can get soil carbon right. None of these are really grounded in any sort of real data. And so if anybody has any great ideas to do this for me, I would love to chat with you about it. And please cite this or read it. A lot of work. If you need a copy, I'll send it to you. I have the PDF. But despite that lack of data, there was really great work done by Sia Sho, Nina Wurzberger, and Ben again, and Caitlin Hicks-Priest, where they worked together to put in explicit ectomycorrhizal fungi and AM fungi into corps. And so they were predicting how much of net primary production was going into ECM fungi and AM fungi. The ECM fungi were able to mine nitrogen from soil organic matter, where the AM fungi were predominantly only able to get nitrogen from inorganic N. And so this is a really great model. It's really cool. I'm looking forward to chatting with Ben about it and kind of tweaking it back into the larger Fund Corps framework. But what they were able to do is they were able to look at how differences in ectomycorrhizal traits impacted competition between saprotrophs and ECM fungi. And so here they had different kind of trait spaces for the ECM fungi. And these are all just depending on where they're getting their nitrogen from. So whether they're mining it from the easy carbon, complex carbon, or necker mass. And you can see what's awesome about a model is you can run all these simulations and be like, okay, it's these traits where we get an inhibition of saprotrophs. It's here where we get a decline, or we get these increases in decomposition where the ECM fungi actually promote saprotrophic activity. And we can also say, like, here's the space where they're competing. So again, it kind of allows you to kind of build hypotheses that you can then test with your empirical data. And I'm looking forward to kind of moving this forward with Ben. The other challenge that we've had, and I've wrote this into multiple grants, that I was going to use omics data to improve our ability to predict the soil carbon and nitrogen stocks and how they respond to global change. We're still working on it. That was like, I wrote that in the first grant like 7 years ago. And so this is work I've been doing at Ember Morrissey, and she developed this new framework called the Carbon Acquisition Ecological Strategies of Soil Microorganisms and Associated Carbon Pools. It's a mouthful, but all it basically is Ember decided that we could classify microbes by what they like to eat, and conveniently what they like to eat are the carbon pools that are simulated in corpse. And so we have first-order decomposers that like to eat complex plant detritus. We have second-order decomposers that eat microbial necromass. We have passive consumers that eat dissolved organic carbon. And then we have predators that eat other microbes. We didn't put the predators in yet. Ben does have a model that has predators, but I don't want to deal with Lacca-Volterra dynamics right now, so it's not in there. So we worked to kind of get these three groups into the model. And so I worked with one of Ember's students, Jeff Walkup, who's again now at UC Irvine. And he was able to put these three different, so we increased our species richness from 1 to 3 in our model. So we had 1 microbial group, now we have 3 functional groups. And what it means is that it doesn't mean that the first order decomposers only eat complex plant detritus. It just means that they're more advantageous at getting their carbon and nitrogen from there. They can still take up that from other pools. And so Jeff did a really great job of getting that into the model. And so what we did is we then spinned up one of our forest sites in West Virginia, the Furneaux Experimental Forest, and then we ran a really simple experiment. And so we wanted to see, okay, if we change the litter complexity or the inputs to the system, do we get changes in the distribution of these 3 microbial groups? And here you're looking at microbial biomass carbon in the 3 groups, and lo and behold, when you increase litter complexity, decrease it, or decrease it and jack up the rhizospheric carbon flux, you see we get these dynamic changes in the distribution of these functional groups. We then were like, okay, let's see if we can get this model to predict some of the responses that we see to nitrogen fertilization at our site. So we had 30 years of nitrogen fertilization to this forest as well. And one of the things we saw is that we got an increase in soil carbon around 1 kilograms of carbon per meter squared. When we threw the base fund corpse model at it, we couldn't get this response. So if we just turned down exudation, it wouldn't work. And so we needed to change the microbial functional capabilities. And so Jeff tested 3 different hypotheses. One was that we had a community shift that favored the other microbial groups over primary decomposers. One is that they had reduced carbon use efficiency. The other is that potentially they had decreased function. And then finally, because we're in model land, we can test multiple hypotheses. We combined 1 and 3. And so here what you're seeing is that it was when we had that shift in the community as well as a decline in their function that we were able to get that soil carbon gains that match the empirical data. And more importantly, what I was really excited about is across all of these simulations, one of the common responses you see to nitrogen fertilization is that it decreases microbial biomass in forest soils. You see across all the simulations we're getting that, and we're getting those changes in the microbial groups. And so we're looking to move that forward. This is really awesome work that has come out, some of the work that JP and also Owen Brody and others have worked on, where they've been able to take microbial metagenomes and use that in a predictive model using energy modeling. And I was talking to JP about this. I don't understand it enough, so I'm not going to, but it's just really cool that they're able to model individual microbes and how they're impacting rhizospheric carbon and nitrogen dynamics. And so where do we kind of go forward? And so one of the things I think some of these model data needs that we have are we need to kind of continue to find novel ways, because folks in here generate a lot of complex data, terabytes of data. Well, I need like 3 numbers. I need you to put those data into like 3 numbers. And so can we get that into simple frameworks that can help us say, all right, this is how much carbon a microbe spends, what it does with it, and how it impacts its resource capture. And again, this will allow us to move beyond parameterizing microbial traits by getting other things right. Like we might be getting the right answer for the wrong reason. And then finally, I think, as we move forward, there's a lot that we can learn from complex models at small scales that we can then take some of that knowledge and put it into larger Earth system models as we move forward. Did I go really fast? I think I did. All right, so, I think we went through all of this. We journeyed along with me. You saw really bad pictures of myself. And, I think we're at a point where the tortoise, the models may be beating the hare. We might be no one's getting, this joke's not landing. Jenny just shook her head. All right. My graduate students didn't think that joke would land either.
Question?
I blazed through a lot of models. Yes, Jenny?
So, this is great, and to see the evolution. To see the history of this, of the field and of your work. It's just so great. And you're the only one who really knows how it works. So it's awesome. I wonder, to parameterize and even validate the models that you want to build, that you see in the future, do you need tracers? Because my sense is that you do. You really need tracer studies. Other types of data are not going to get you the numbers that you needed. Would that be accurate?
Yes, so we've done some work, doing soil incubations in the lab, where we'll put labeled litter in a jar and see where it goes, and where does it go into particular organic matter or mineral associated organic matter. And my graduate student was like, "It's a jar study. Why are you going to use that to inform your model?" I was like, "Because we have no data." And so we can use that to develop some data. And if we can do those types of, if we can look at the fate of soil, litter and inputs in the field, if we can look at the fate of microbial necromass and where it goes in the field, that would be really important and really valuable for the model, because we can see if we can get those fluxes right. In the back, JP. That's fine. One, I think how much carbon is being from a plant going to an ectomycorrhizal fungus across different environmental conditions, and across, if we could get more than a couple of numbers, like Hochberg, juice in a tree in Europe and seeing where that goes. If we get real spatial and temporal variability. And I don't know how you do that.
That's doable.
Yeah.
I can tell you 3 other ones that I don't know how to do. How do you measure microbial biomass and actually believe the numbers? How do you measure cell size? Seems like all those things are.
Yeah, they are. All right, so you kind of set me up, but that's the one I like the most. No, but yes, that's what I mean. If I can use a catch-all, it's like, how does the microbe spend its carbon? And you're doing some of that in the work you guys have done, but we can make it simpler for these, that's great at the small scale, but if you're going to put that in a larger Earth system model, you need 3 number, you just need fractions that scale with environmental variability. Yes, Corinne.
Thank you. This is really helpful also just to understand. Are you or people thinking at all about incorporating different relative abundances of functional yields into model, whether that's just EMF versus SAPs, or even different functional
Yeah.
bacteria?
Yeah.
It seems like,
Yeah, so I probably went too fast. But if you go back here, past that joke should have landed. I'm sorry. So the great thing about what Ember proposed is that she can measure the distribution of these groups, or who these groups are, using quantitative stable isotope probing. So she can add complex plant detritus and see which microbes actually like to eat that. And we can kind of get a distribution. And then Ember tells me that some of these traits are phylogenetically conserved. And so we can kind of take that data and make it broader and bigger and scale it up. No, we can do all, you can make necromass, labeled necromass, and throw it in the soil. You can add in DOC. You can do all of these different things. There have been folks, so Will Weeder has the Mimics model that has copiotrophs and oligotrophs. Even if you go back to, blanking on his name, he's at Toledo. Anyway, he had, sorry? Not Mike, it was the older guy. Daryl Moorhead, he had functional groups that decomposed litter. He had ones that did complex plant detritus and one that did other things. So it's trying to find what level of complexity you want to add without making, you don't have a gajillion parameters that you're over parameterizing your model. And if you want to scale up, you don't want to do that. People get mad at you. Because they're like, why are you going to do that? It's going to be way too complex when you go to an Earth system model. But yeah, we're trying. Yes?
Question about like the semantic use of microbial explicit, because you just said something that I did not know, and I think I missed it. So I guess I always thought that if you were concepting a microbial explicit model, it just had microbes in it. Like it's not Century where there's kind of an implicit microbe with a lost term or something. But you said it had to be non-first order kinetics to be microbial explicit. So tell me more.
Yeah, so Century has a microbial pool. Descent has a microbe pool, but it's just, well, Bill Parton and Melanie have worked to make that different, but traditionally it has a microbe pool because you need somewhere to put that carbon and nitrogen. And so microbial explicit means not having first order decay, because the microbes are the K, right? And you move that K around based on soil temperature and moisture conditions. So when it's wet and warm, K is higher. When it's cold and dry, K is lower. No, yes, and so what you have in the microbial explicit models is we'll go to, sorry, this is further than I thought. I shouldn't have done this. Here we go. So you have microbes that actually die and turn over. You have microbes that take up dissolved organic nitrogen. They die and it recycles. And more importantly, they make enzymes. And those enzymes work through Michaelis-Menten kinetics to break down soil organic matter. They have carbon use efficiency. Like, it's not perfect. Like, it's not at the level where we have leaves, where I sort of get angry. Hopefully no one's a photosynthetic modeler in here. But they got it like 90% of the way there, and they're fighting over the last 10%. We're maybe, I don't even want to estimate where we are. But we're not where they are.
Great.
All right, well, thank you.
Thanks, okay, everybody. Thank you all for sticking around for the last talk. We have had an incredible lineup of speakers today, and I am really, really excited to cap it off by introducing Dr. Jennifer Pettridge for our final talk. Dr. Pettridge is a senior scientist at the Lawrence Livermore National Lab, and she's the lead of the carbon uptake pillar and the environmental isotope systems groups at Lawrence Livermore. She's also the sustainability theme PI for CABI, and she has been at least an editor for what seems like almost every single major journal in soil sciences and biogeochemistry. So a huge service to our community. If you have personal grievances about her decisions, see her afterwards. You can measure somebody's sort of status in the field in a bunch of different ways. The way I sort of think of it for Dr. Pettridge is that you've heard now several times today people that she's close with affectionately refer to her as JP. And I feel like the ideal trajectory for a career in this field is like you get into graduate school, you publish your first lead author paper early on, eventually you'll get into the National Academy. Somewhere in between there, at least based on the people that I really respect in this field, you become sufficiently familiar with Dr. Pettridge that you can start calling her JP. So that's sort of where it is. And full disclosure, my ulterior motive in inviting her here was that we could all get in on that group. There are other ways to measure someone's status in the field. The Ecological Society of America last week decided maybe a more appropriate way would be to name her one of the ESA Fellows. Congratulations. That follows selections from the Geochemical Society's endowed lectureship in 2019 and 2021. And she's also won the U.S. Department of Energy's Ernest Orlando Lawrence Award. And I want to note why they gave her the the Lawrence Award. It was, in quote, for her pioneering work in quantitative microbial ecology and leadership in developing and applying isotopic tools that help us discover, and here's the important part, and quantify how changing climate shapes the role of microorganisms and plants in environmental biogeochemical cycles, right? So this is exactly what we've been talking about today. This is a perfect description of why Dr. Petridge is so important to our field and why we are so excited to have JP here at this symposium with us. So with that, I'm going to turn it over to Dr. Petridge.
Well, thank you, Ben, for having me. And yeah, everyone can call me JP. It's not a club. I've been called JP since I was a kid because there are a lot of Jennifers born in the 70s. Let me just first make sure that you can hear me. I think I turned it on. Okay, great. So I can't say this is a culmination, but there are some threads for sure that you've heard about earlier today, and particularly in Eddie's talk just now, and even in some from Colin's last night, that we're going to pick up on here. I thought it'd be fun to try and tell a slightly more cohesive story than I sometimes tell. So Fungi and Friends, that's cohesive for me. I'm not good at putting up outlines because I would never stick to them. But essentially, we're going to start big scale, and then go really, really, really small scale, and then we'll go back to the big scale. So there's your outline, okay? And I want to start by thanking the people who really do the work. And there's probably more that should be listed here, but a couple of these folks, Peter and Rachel, are here in the room, and I want to just give them that shout out, and I'll try to acknowledge folks as we go along, because at a national lab, kind of our hallmark, if you saw Oppenheimer, is team science, right? We don't do anything on our own. It's a somewhat different structure than academia, and everything, it takes a village. So, moving forward, I think of this planet as being run by the microbes. That's my bias. Every biogeochemical cycle, for the most part, has the bio part has a microbial element, and climate change is impacting how those organisms exist, where they're active, where they're not, and the processes that they drive, and particularly greenhouse gas emissions is the key one we think about, but many of the other, the resilience, the pathogenicity, all of these things affect the roles that they provide to ecosystems that we live in. And so, yes, we need to get better at measuring them and really modeling them in a more than just let's model what we already know to be true, but in a predictive way. We need to be able to model the future that is coming. And this is a paper that figure from Will Weider's paper from some time ago that I still just really enjoy, because it really shows you how wrong we can be with our models. A traditional soil model, if it doesn't have that microbial explicit element that we just heard about, can really give you a very, very different answer. And I can't tell you that one is right and one is wrong, because we're not even quite there yet. But that's true whether we're just talking about decomposition or whether we're talking about adaptation to warming. We're relatively close here to Harvard Forest, and my dear colleague Kristen DeAngelis and others have shown over quite some time that warming effects on microorganisms that live in soil doesn't just head in this direction. It goes that way for a few years, and then it jigs and jags, and the answers have been complex, both for who's active and the overall effect on carbon cycle and carbon pools and nitrogen pools. So just because we know something today doesn't mean it's going to keep being true in 5 years. So I think that's important. A little plug for this kind of nice colloquium report that I and some colleagues wrote a couple of years ago during COVID, that just really tries to substantiate why I think we do need to think about including microbes in models. And obviously not every single taxon is going to make it, but we need to think of ways of aggregating their demographics, really, when they grow, when they die, what they do. So what drives me, in terms of the kind of higher scale biogeochemistry is this imbalance that we have on our planet that has to do with carbon. First of all, we've got too much carbon in the atmosphere, and we've lost an enormous amount from our soils, something on the order of, now it's about 500 gigatons. Those are billions of tons of CO2 equivalents. And a lot of that's because of microbial activity. A lot of it's because we plowed up almost half the planet, terrestrial land surface of the planet. And And when you do that, you put dinner and lunch and breakfast in front of microorganisms, right? And they're happy to respire it back to the atmosphere. A lot of it's actually eroding away, and that's not necessarily ending up in the atmosphere. But we really got a debt to our soils. I think of it that way because that carbon does a lot of good, the organic carbon, once it's back in the soil. And a lot of the work that I've been collaborating with colleagues on in the past decade has been, can we put it back? And probably first, before we can start doing that, or at the same time, because we can't wait, honestly, we need to understand how it got there in the first place. Why does it persist in the soil to begin with? So I want to just, this is a plant and soil rhizosphere-oriented audience. You probably don't need to hear any of this, but I want to make sure we're on the same page that most soil carbon is derived from roots, not from plant litter, above ground being. That decomposition rates tend to be much higher in the rhizosphere and in the microhyphosphere. We have a lot of different biogeochemical functions, higher rates of proteases, chitinase breakdown, just everything's kind of spinning faster. And that bloom of organisms that is driven by exudation and turnover of root tissue is driving wonderful micro food webs, right? We've all tended to focus on whatever we focus on. We focus on bacteria, or we focus on AM, or recto, or viruses. I'm trying to get myself and you to think about those trophic networks. And because really, if we want to be able to predict the sum, we've got to think about how they're all eating and sharing and interacting with each other. And then finally, interacting with the geophysical substrate that they're within. And that's become more and more interesting to me as I'm working in this field of carbon dioxide removal, trying to literally suck CO2 out of the atmosphere. Well, in soils, one of the most exciting new technologies is the idea of adding basalt, crushed rocks that will naturally weather and have these reactions that are pulling CO2 out of the atmosphere. Well, what if we could encourage that with particular biota? What if fungal exudation of organic acids made all of that happen faster? That would be a wonderful part of the solution. So I also want to tell you that, yes, the roots are soil carbon, but they go through a microbial lens. There's some who will argue that there's some amount of root carbon that actually directly sorbs onto minerals, and I'll give you that. But there's a lot of carbon that goes through the biomass of bacteria and fungi, and yes, even viruses. Even though they're tiny, they're a lot, and they add up. And so this project that I lead called Microbes Persist is really all about looking at, these are some bacteria on a little goethite surface, and this is a fungal hyphae, and it's plastered with little, I think these were kaolinite particles. But you can just start to imagine the scale at which these phenomena occur. But all of that biomass adds up. And does it stick around? Is it just sticking around because it's created quickly and decomposed quickly, but there's a lot of it? These are things that we don't fully understand. One thing that we'd hoped, Eddie mentioned the modeling paper that we just published, was that maybe we could just measure the right traits. Because above ground we can do that. We can measure NPP, and we can classify things as trees and forbs and deciduous, and we can put things in boxes and think about their demographics, and those ecophysiological lifestyles help us predict how those organisms exist and interact with each other. And we thought, oh, well, all we have to do is be able to do that with microbes, and then we'll be able to predict things like carbon cycling. And so we came up with a big long list, and you don't need to look at the details here. There's a whole bunch of ideas of the different traits that we thought maybe we could measure, especially now that we've had this sort of omics revolution, and we can measure a lot more things than we used to be able to measure. Much of it with DNA and RNA and proteins and metabolites, and some of it even at a taxon-specific level. And we thought, okay, we just got to, go through the list, pick out which are the top 5, and then we'll have it sorted. And that was 6 or 7 years ago. I feel like we're kind of getting further from being able to do that, to be honest. But if I were to just say to you, what traits do you think are likely to predict soil carbon cycling or soil carbon pools? What would you guess? CUE, good. That's a great one. We tried that one, and sometimes it does. Depends where you are in the soil. Well, actually, so this is a rhetorical question I like to ask because then I try to have this big denouement where I say, nothing, and we can't find anything that correlates. But Colin messed me up last night because he showed us that the fungal community composition actually does predict forest soil carbon. So that's cool. That's one. There's relatively few examples of correlations like that. I think you also said that when you tried it with the bacterial community, no good. So I'm still on my track here, which is the point being we don't have a great capability of using these traits that we measure, things that have to do with phylogeny, if it's 16S and ITS, and using that to predict functional attributes of the system. And this is one particular paper, but I could show you there are nearly a dozen where folks have tried to do a meta-analysis of particular types of genes or even things like CUE, and they, on the whole, don't do a great job, which is a little frustrating because I believe there is a relationship. So we must be doing something wrong. And I'm going to argue that what we do wrong, as it were, or what we could be doing better on, are how we conceptualize and how we sample. And the first being that we tend to sample with a shovel. The first being that we extract whole community molecules, whether it's DNA or RNA or peptides, if we're in the sort of community ecology space. So you're scooping up soil or a root or even an infected root tip, and you're extracting from that. And what that's doing is smearing together a lot of what we call relic DNA. So this is DNA that's actually extracellular, that's just from organisms that croaked yesterday or maybe 100 years ago. We've actually shown that with 14C radiocarbon analysis that some DNA is very, very old. So everything we were taught about, oh no, it's degraded immediately, there's nucleases in the soil, not really, not after it's sorbed to a mineral surface. There's also gobs of organisms that are just dormant, that don't know what they're doing. I mean, yeah, Jenny, you sort of alluded to this. Spores, they've got all sorts of different ways of weathering moments they're not keen on. And what we find with our isotope tracing is that often only 8 to maybe 20% of the total community is active, growing. What's everybody else doing? But the point is, if you're sampling that whole crew, yes, that gives you some indication of the future of that system. But if you want to predict today's CO2 flux or today's carbon storage, you got a blurry picture because you're sampling too much, as it were. Okay, the other thing that we tend to do, it's related, is that we tend to scoop up soil with a shovel or trowel, but something that's bigger than tweezers, which is really probably the best way we need to be sampling, because these organisms live at scales that it's hard to even wrap our heads around, other than if you spend a lot of time with an SEM and you're really looking at that micron scale. So they live in these habitats that we smear across if we're just scooping up rhizosphere and mixing it with hyphosphere and bulk soil and the interior of an aggregate and the exterior. And then I think I kind of made the point already that we tend to study only one kingdom at a time. So relic DNA, a real problem. Noah Fier and Paul Carini published this some years ago. We've replicated it and showed, yeah, it's a real problem. 15 to 30% of your DNA extract could be just DNA that's out of the bag. It's just sorbed onto minerals. You're extracting it, but it's not necessarily from an organism that was intact and thus viable. So that's one reason why we have really focused on stable isotope probing. And isotope probing, I like to think of it as a kind of a basket of techniques. Originally, when it was developed by Ratajkowski et al., it was really specifically about adding a substrate that was enriched in a rare but stable isotope, looking at the incorporation into DNA, and then doing some sort of profiling of that community. And that's great, and that's been improved upon, particularly by the Hungate group at Northern Arizona, where they've added normalization to qPCR to make it even more quantitative and get you to the point where you can actually look at a taxon-specific growth rate or mortality rate even. But there's other ways to do isotope probing writ large, and that for a long time we did it with microarrays. We would sorb RNA or DNA to oligonucleotides that had been tethered to a microarray surface, and then we would stick it in this cool instrument that we have called a nanosims, which is an imaging mass spectrometer, and we could go and look at which spots were enriched. It's just a way of kind of doing a quick analysis, but taxon specific. Now we do that with SIP. We can also do it with the whole organism, and again, not everyone has a nanosims. Harvard had one. They're about to decommission it, sadly, but anyway, there are more and more around the world, and you can apply for time to use them at various user facilities. But this can tell you about, what's the enrichment of a particular nematode, of this fungal hyphae, of individual bacteria, and yes, even viruses. So lots of different ways, and you can start to kind of layer on other techniques where you might be using a beamline to look at molecular characterization, or you might be using even an accelerator mass spec to look at the longevity of particular molecules. So this family of approaches is something that kind of underlies the majority of the work that goes on in the groups that I work with. And more and more these days we're doing it in whole systems, meaning we've got a plant there as our pump, and sorry, I know that sometimes is a little rude, but we are using that plant to take enriched 13CO2. We fill the box with very expensive CO2. The plant does its job, and the carbon is put below ground. But as all of the diverse metabolites and signaling molecules and VOCs that one would expect, and it's not going to be just a single compound. And then everybody who's excited about exudates or VOCs or whatever it is as a substrate can take that up. And we can do that both in the laboratory setting with nice controlled systems, but also somewhat controlled out in the field as well. But with, the wonderful thing is when you do it out in the field, you tend to get a wider complement of the food web, right? Nematodes and ciliates and some of the larger things, they don't do well when you bring them in the lab, so they need to be out in the wild. Okay, so that's using 13C. There's lots of 15N ways to do tracing as well. I don't think I'll talk about any of that today. But the other newish approach to stable isotope probing, or SIP, is to use 18O heavy water. Everybody likes water. Everybody's growing is going to take up water. So it's the sort of universal way to label who's active. And yes, there are organisms who are not dividing, who are still, you might call them active. I'm going to not call them active today. I'm sorry, but that's just a way of us querying who's getting on with creating more of themselves. So, and if you know anything about SIP, you know it's a grad student killer, right? It's painful. Oh my God. We used to, if you've done titrating, right? So this is like that, where you're literally counting drops. You count 8 drops, you move the 96 well plate, you count 8 more drops, and it takes days. And it's really painful. We've figured out how to train a couple of robots to do that. And so now we're doing high throughput SIP, 16 to 24 samples a week. And maybe that doesn't sound high to you, but it's high. So that's an exciting thing that not only our lab has now going, but the colleagues at the Joint Genome Institute offer as a service. And we actually, I should say, learned this from colleagues at Cornell. So hopefully more and more will bring this on board. Okay, back to habitats. Lots of different habitats in soil. Often when I'm giving kind of my rhizosphere talk, I go into the differences between these different zones. I think of many of them as being driven by the biology, others being driven more by the sort of physical attributes or mineralogical attributes of that zone. But today I'm going to focus mostly on what we're going to call the hyphosphere, or the zone surrounding mycorrhizae. And I'm mostly going to focus on AM, but there's a little slop there, so I embrace that hopefully the mechanisms I'm talking about are true for both AM and ectos for the most part. So you've no doubt, if you study or think about mycorrhizae at all, you've seen some of these wonderful videos. This is particularly one from Toby Keir's group in Amsterdam showing internal movement, a highway of movement, and the idea of a resource economy, of the plant is sharing with the mycorrhiza is bringing nitrogen and phosphorus and even water back to the plant. And I couldn't get the video to work on this one, but go look it up. Bacterial highway, literally using the surface tension of these hyphae as a way to cruise along. And we don't really know that that happens in soil. These are fairly clean images here, but presumably there is some amount of movement that's afforded by these long lateral surfaces. And that is just evocative on its own. But when we start to think about the amount of photosynthate that can be allocated to these structures, and it's up to 30%, and I've seen numbers that are even higher, but that seems like a fairly conservative number. What are the system level effects of that sharing of photosynthate with this network? Well, the first thing I like to think about is the work that Craig C., who was a graduate student of Peter's, did when he came to visit us for a little bit, where he did a thought experiment of not just the NNP that's being shared, not just the water that's being shared, the signaling molecules, all that sort of thing, but just how much is there? What's the volume of this stuff? And if it's fine roots, you think about a cubic centimeter, you've got something on the order of maybe 7 centimeters of fine roots per little cube. If it's AMF, 2,000 centimeters of length within that little volume. And if you include all hyphae, Craig, this is back of the envelope, but Craig estimated something on the order of 100,000 centimeters. That's a lot of surface area. Because what that's saying is our definition of the rhizosphere, where we think about the interactome as being maybe, a couple millimeters at most away from the root, is actually like the whole dang thing, because these things are going out. I mean, in an infected plant, it's just a much bigger volume of the soil that is being influenced and spoken to by the plant, as it were. And so that's something that I don't know, has really stuck with me when I think about all the phenomena that I'm going to continue to tell you about. So how do we really pinpoint the effect of the hyphosphere, the zone surrounding these mycelium, versus what's going on in the rhizosphere? Maybe it's all just a continuum, it's all a bunch of pipes, or maybe the phenomena are slightly different. Well, the way that we've tended to, we and many others have tended to do this is to build pots in the lab like this one, where you have a slight air gap in the middle. So you've got a plant on this side that's been inoculated, and there's a little bit of mesh here that the roots can't cross, but the hyphae can. And so you get a no plant side that's just influenced by hyphae. And then you could do fun things on that side. You could add 18O water and see it going back to the plant, or you could be measuring just the carbon that the hyphae are contributing. And similarly, if this is, I'll just tell you, Anne Kekuritis had to do this 6 times before she could actually build these that worked in an experimental sense. Rachel Hestern made this design where we had a single pot, but mesh cores within it. Again, accomplishing largely the same goal, in that we could distinguish the hyphosphere soil that was under the same sort of water conditions and temperature and all the rest, but distinguish it from the soil that just roots were influencing. But these kinds of experiments that the two of them and also Rachel Neurath have carried out in our lab have really allowed us to dive in with isotopes and really target these structures and what they're doing and who lives among them. And we've used some standard techniques like staining to also just look at inoculation. But the wonderful thing is that sometimes you can actually just see them. And this was, I think, taken with an iPhone. It should be on a billboard, right? It's like amazing just to be able to see how they're actually clogging up little particles together and making aggregates. But anyway, I digress. A lot of that is also we image in our nanosims, and this is in the core of this little, structure here is a hyphae that would have been attached to a root, that would have been attached to the above ground parts of a plant in a 13 CO2 chamber. So the bright colors here are showing you isotope enrichment making its way out into those hyphal structures, stuck to minerals. This is the definition of MAM, mineral-associated organic matter, in my mind. I guess I'm one of those people who needs to see it to really believe it, but that's, yeah. So when Anne looked at some of the data from a study like this, where she looked at just the no-plant side of a pot that had been inoculated with Rhizophagus irregularis, she found that there was quite a lot of the 13C coming over that was making it into what we often call the light fraction. This is particulate. It tends to be somewhat more ephemeral. It tends to turn over fairly quickly. But overall, we saw an increase in the total amount of carbon on that side of 2% in a matter of like a month. Now, that, I don't know, that's a big number in the carbon world. I'm not saying that 2% is going to persist indefinitely, but just that shunt it's an important assessment of the ability of the plant to move that carbon. And I'm going to say, Eddie, it's starting to head in the direction of the numbers that you wanted, albeit not with an ecto. But we also saw something on the order of about a quarter of that carbon, was sorbed to mineral surfaces. And why do we care about that? Well, some data tells us that once you get organic matter sorbed to a mineral surface, it's a lot more likely to persist, to hang around for the long term. Now, what are they sharing? What's the stuff? What's the magic, goo that's what are the exudates, essentially, of these hyphae? I wish I knew. We're really working hard on that because we're getting to the point, right, where we can distinguish root exudates from just DOC that's floating around in the soil system. But this is the best I can tell you today, which is we did some 13C NMR of the soil with the hyphae and then soil without. Sorry, this is the without, this is the with. But this one peak where we think there are carbohydrates in particular, it's an O-alkyl kind of carbon. We think you could call it sticky goo if you want to, but it's just a kind of exudate that we think that these hyphae are actually producing on purpose for the same kind of reasons that roots make exudates. No idea. If anybody knows the answer already, great. And if we don't, we should all be working on this because it seems important. But I'll move forward to a more complicated experiment that Rachel Hestern, who's here with us today, ran when she was working with our group as a postdoc. She's now faculty at UMass Amherst. And the question there was, well, okay, AMF do this, but do all mycorrhizae do this? So let's try a couple different kinds of beneficial partners. And we settled on looking at an arbuscular and a sebastanoid. So sebacina is more like an orchid fungus. Some folks would not like calling it a mycorrhiza, but it walks like one. So, and how then does drought affect, like an environmental stressor, affect their ability to take carbon from the plant and move it out into the system? And does one act more as sort of a quasi-saprotroph, or are they all just acting as symbionts? And so when Rachel looked at how much carbon was moving out into the hyphosphere, she actually did SIP on the microbial community, largely bacterial community, surrounding the hyphae of these different kinds of mycorrhizae. And so the data that you're looking at here, ASVs, taxa, species if you want, whatever, and then APE is atom percent excess. So that's essentially telling you how much enrichment of the rare isotope above a background. And quite interestingly, we're seeing that in both of the inoculations, we're getting more than in a non-inoculated, and no statistical difference in terms of the community enrichment. So they're both helping the community take up that carbon fixed by the plant, shared to the root, given to the mycorrhizae, and transported out into soil. And it really didn't matter what the different mycorrhizal type was. Now, one thing that was really exciting about SIP is, again, I kind of alluded to this earlier, it really helps you hone in on who matters. So she also did some SIP, I mentioned earlier, with 18O water, just to look kind of more ubiquitously at who was growing. And when Rachel first looked at the 16S data, or early on at least, This is a plot of all the 16S data before we'd pulled out just the enriched organisms. And I don't know, this is what made me cry in grad school when you see all these overlapping ovals and you're like, I worked really hard on this and there's no statistical differences. But the exciting thing is when you look at just the enriched organisms, when we've used the SIP data to tell us who's actively growing, there's like this wonderful separation between the treatments as one would expect. And Rachel has since published this and looked particularly at the drought effect, which is quite remarkable in this system. So water limited is code for drought. We're not allowed to call it drought because it's a soil that naturally experiences. So anyway, but more water, less water. And you can see if you don't have the inoculation, there's a really big difference in terms of the ability of these organisms to grow. That's essentially what you're seeing on the axis here. But when they've been inoculated, and again, it doesn't matter whether it's with the AMF or with the Saponicina, they seem to be sort of rescued in their ability to keep growing surrounding these hyphae. So maybe it's that special sauce, that sticky goo or whatever it is giving these organisms the wherewithal to continue even in the face of a water limitation. We don't fully understand the mechanism there. That's something I think, well, maybe Rachel will keep going on that. So who is it specifically that's living in the hyphosphere? Well, I'm not going to read all their names to you, but I'll just tell you we did dig in deep using a couple of different phylogenetic profiling approaches. And first, just to show you sort of the standard ordination plot, that in the blue, you're looking at without an AMF, and in the orange, you're looking at with. And you can see there's a distinction. This is without any SIP. And when we used a technique called DE-seq, where you look at the relative abundance of organisms, or their sequences at least, you can see that there's a series of organisms, even if you can't read the text here at all, that are being promoted. There are more of when they're surrounding a hypha or mycelium, and then there are some that are, I can never think of the right word, but deeper. There are less of them. So you get a discrimination effect, which makes sense. We absolutely see this in the rhizosphere all the time. And it's clearly affected. In this case, it was by an AMF. Interestingly, when we added SIP, we were also, in that case, doing metagenomics. So we were looking at shotgun sequencing and assembling what we call metagenome assembled genomes, or just putting the puzzle pieces together with sequence data. And in that case, we saw quite different patterns. It was different organisms and less driven by abundance. And I'm just going to pause here and say, this is a bit of a cautionary tale about amplicon versus shotgun. I think that much of the field that at least that I exist in is moving wholesale into this shotgun world. Like, oh no, more data is better. We want to know more about functionality, and that's what we get from metagenomics. True, but we tend to get much less depth in terms of the coverage of each individual organism. If we're lucky, we get 300 to 500 mags that assemble for a given experiment. Whereas any kind of amplicon sequencing, you likely know, we often get many, many, many thousands. I think if you think about a rarefaction curve, we're sampling the total community better. So there is I want to just say that's a trade-off. If you're trying to characterize a community, save your money. And just sort of the who's there, and I still vote for the amplicons. Of course, if you want to find the magic organism that can do just that one thing, then I, of course, am going to recommend doing metagenomics. And that is what Aaron Nuccio and colleagues went after, again, surrounding those little tiny threads. I showed you that iPhone photo. They literally took tweezers and plucked out as many of those as they could. I think Aaron, in the end, ended up with something like 300 nanograms of DNA. That's a small amount to put into a SIP gradient, but she managed, and it was only enriched by 1.5%, which in the tracer world is not much. But she managed to get a SIP metagenome to work, and the SIP metagenome showed her essentially who the organisms are who were particularly enriched. This isn't everyone, but we're going to call these key organisms because they're, the dotted line here is how enriched the hyphae were. So we had some organisms who were actually more isotopically enriched than their substrate. That's trophic theory right there. But in particular, it's this Myxococcata. It's known as a facultative predator. When we looked at its genome, it has a lot of protease genes breaking down organic matter. We also saw some fiber bacteria and some verruca microbia. They both had genes for breakdown of N-acetylglucosamine, otherwise known as chitin, which is what makes up many kinds of mycelium. And they also seem to be targeting glycoproteins. That's what we actually think the sticky goo is some sort of sugary protein molecule that is, again, I can't say if it's being purposefully exuded or just sloughing off the exterior. And then, intriguingly, actually a nitrososphere. Well, this is an ammonia oxidizer. Ammonia oxidizers don't live on organic carbon. So that was a bit of a head scratcher. They live off of CO2. They're autotrophs. So there's actually this food web going on where there's enough turnover of the organic carbon being put into a 13CO2 space that these organisms are able to take it up and yet be enriched enough that we found them and were able to create a mag. But that's still a bit of a puzzle. These tend to be very slow-growing and rare organisms, so we're quite amazed that we found it. But altogether, this little crew we think of as a very small food web that lives in a very small space that you'd need to sample with tweezers. So, getting even smaller, and this is going to be the nadir, as it were, of the talk when I said we were going to go to really small and then come back up again. We also have other experiments, these more done in the rhizosphere. Evan Starr took some metatranscriptome data and showed we had an enormous diversity of viruses, RNA viruses. And RNA viruses, lots of them are known to target fungi. So it's not just these fungi sharing with their bacterial friends, they're being attacked. And viruses as predators, of course, that makes sense. We've all, well, maybe not everyone had COVID, but anyway, we know about that. But I think, how do we model them? How do we think about them as different from the Myxococcata, which tends to just engulf its prey? But I think we got to include them in our models, Eddie. And you said you hadn't got the predators yet, but this adds an even another twist. And we've shown they actually do follow the Lotka-Volterra dynamics, but very, very quickly on the scale of hours. So that's all I can tell you about all the pieces of the puzzle that are in both the hyphosphere and rhizosphere in terms of food web. So now I want to kind of go back up, maybe eventually to the global scale. All of this sharing and killing and organisms interacting, what's the fate? What's the sum result of all that in terms of where is the fungal carbon going in that great big interactome that I showed Craig C. had calculated? Well, first of all, lovely study that collaborated on with Peter Kennedy and his postdoc, Francois Maillard, where we're actually looking at different qualities of fungal necromass. And Peter's group has gotten very, very good at growing essentially Melonomyces bicolor, and I think it actually has a new name now, and I'm sorry, Peter, I can never remember it. I'm sticking with this. Okay, apologies. But you can grow essentially the same taxon in conditions where it produces a lot of melanin and conditions where it doesn't. So you got a white form, you got a black form. And they've showed it many times, but as expected, when you have the high melanin or the black form, you get much less of decomposition, and so it's like any kind of litter bag experiment. If you've done one of those, you have quite a bit remaining. But in addition to this, of course, we added isotopes, right, when we grew it. So these are expensive necromass. And we put it out in little packets out in the field. We call them coupons. I don't know why, but we put them out and let them, like a litter bag experiment, and let them decompose over the course of part of a summer. And then came along and scooped up that material and did stable isotope probing to look at who was decomposing the dead necromass, the leftovers of this fungal material. And the answer is kind of everyone. It's, there's a little bit of a trend in that, there was the white material, the less melanized, was more yummy. But really, we saw a lot of diverse activity. And it could be that there's cross-feeding because these were out for several weeks. But it seems that there's quite a lot of organisms who are happy to act, and sorry, I should be saying these are bacteria. Bacteria that are acting in a sort of saprotrophic manner, and probably some other fungi as well, that are happy to be gobbling up all of that necromass, more so the low melanites. So, point being, it's moving, this carbon's moving through mycorrhizal networks into bacteria. That necromass, once it dies, is then also getting cycled again and again and again. And then some of it ends up, in some magical way, sorbed to mineral surfaces and sticks around for a very, very long time. In another kind of litter bag type experiment, Craig T. Showed that a lot of that carbon was actually getting sorbed onto the surfaces of little mineral particles. And these images are a little bit more messy because they're not just a clean necromass itself. We've got minerals and necromass mixed together. But he hypothesized that he think a lot of saprotrophy is occurring and chewing up that chitin and other materials. And some of it is then getting sorbed to the minerals. And we see that, I showed it to you earlier in some of Anne's data, but we also see it just physically in that we tend to get these associations which are really quite strong. And there's quite a bit of data in the literature also that shows when you have more of these mycelium, you get aggregation. And anytime you could package minerals together so that you get a little clump of organic carbon in the middle like a jelly donut, that's a great way to protect that carbon for quite some time. Some older data I worked on with Marco Kailoit some years ago showed that when we decompose fungal necromass in forest soils, that the amino sugars that are derived from those the N-acetylglucosamine tends to sorb preferentially to iron oxide surfaces. Doesn't mean it doesn't go anywhere else, but it was every time we saw an isotope labeled chunk of organic matter, it was on the surface of an iron oxide. And we see that at even bigger scales. So this is some work that Ashley Lang did, and working with Rich Phillips and myself, looking across all the neon sites, at least those are on the eastern seaboard, and looking at how does fungal necromass relate to soil carbon persistence. And so what she was plotting here was the amount of ectomycorrhizal basal area. You've got a lot of these sites that have both AM and ecto trees, and she was able to find samples that were purposefully chosen from either ecto or AM stands or mixtures, and then look at the proportion of carbon that was associated with a mineral. And again, that tends to be indicative of it lasting longer. And for the majority of the sites, there was a pretty clear negative relationship. Yeah, there's this one that tends to have really anoxic soils and didn't behave. But we see the biggest controller of this relationship between the mycorrhizal, the biota, and the persistence of carbon being driven by, again, whether or not there were a substantial amount of iron oxides in the soil, and then secondarily, whether it was an ecto versus an AM-dominated stand. And we've heard that from several of those speakers today. So, even getting to the global, I want to, you've seen this paper cited several times. Hawkins et al., Toby Kears' group, again, really did a lovely job, and I think it kind of riffs off of what I told you Craig C. had calculated in terms of the interactome. They really calculated it by doing a meta-analysis of, I think, something like 200 studies, showing how much of plant carbon fixation and CO2 equivalents is ending up in AMF, is ending up in ectomycorrhizal fungi, and in ericoid mycorrhizae. And that all told, we're talking about something like 13 gigatons, 13 billion tons of carbon that is CO2 fixed by plants is getting allocated at least temporarily to that underground mycelium. That's a really big number. I didn't give you my CDR talk, which is all about how much we've got in the atmosphere and how much we need to remove and how it all balances out. But let me just tell you, 10 billion tons is the amount we need to start removing from the atmosphere per year by 2050 if we're to keep the planet habitable, to be blunt. So this is the number, this is a kind of big number in the scale of what we want to be seeing. So I think that I'm just going to put out a really radical idea here. What if they could do more? What if 13 is just the the normal place that mycorrhizae are drawing from. What if it was double that? What if on our agricultural soils we had more inoculation, more successful inoculation, more beneficial roles for mycorrhizae in the, again, nearly half the planet that we're practicing agriculture on, and all of that mycelium was getting out into that enormous interactome, and even just a tiny bit of it was sticking around and repaying our debt to our soils. So this is a wacky idea that I and some others are getting funded by the Chan Zuckerberg Institute. You know who that is, right? Go to Facebook. Use Facebook, because some tiny, tiny little bit is going to fund science like this. Sorry, no, I really can't say that after some terrible things that have happened there. But the idea is to use some of our amazing new gene editing tools to not just edit microbes, but edit the whole system, make the plant more photosynthetically efficient, make the roots longer and tougher, and make them better places to be inoculated with not just mycorrhizae, but all kinds of beneficial organisms. Is this a way we could actually stuff more carbon in the ground? Maybe. Maybe we'll learn some cool science along the way. So I will wrap up now by just giving you some take-homes. And the first being that, for the first part of the talk, that I think we've spent a lot of the last, say, century looking at bipartite interactions, plant, pathogen, plant, rhizosphere, fungi, bacteria, and we need to, I think, as always, move to a more holistic place where we're really incorporating the viruses, the protists, the ciliates, the whole gang that's involved if, again, we want to be understanding the outcome, the emergent effects of that ecosystem. And I would also argue that we need to be quantitative. And that's where the isotope tracing comes in. And I'll acknowledge right now that it is expensive, but it allows us to actually measure those fluxes that otherwise we're only measuring net changes as opposed to gross fluxes between organisms. And then I'll just end here. This is sort of the by the numbers, the things that I've tried to show you that I want to kind of get maybe one or two of these numbers into your head. That there's an enormous amount of hyphal length, a huge interactome. That 100,000 centimeters per cubic centimeter is still kind of shocks me. Even if we're off by an order of magnitude, it's still a really big number. That AMF, I didn't show actually all of these details, apologies, but I'll just say that we recently published some work showing that AMF can move up to a third of what the plant is transpiring in terms of its water. So they're wicking it along the edges of the hyphae. Rachel has shown in previous work that they're also able to move ammonium gas, and that can be a substantial amount of the plant's nitrogen needs. We saw significant carbon moving the opposite direction, increasing the soil carbon pool. A lot of that became mineral associated. I described to you the microbial communities, in particular that neat little food web of organisms who are quite key in taking up that carbon. And interestingly enough, those interactomes tend to occur quite some millimeters away from the root. And that's, in our experience with AMF at least, because the mycelium that are quite close to the root are functionally a pipe. They've lost most of their nuclei, and where they're growing and exuding is at the tip, not unlike a root. And then finally, the point I made at the end, that 13 gigatons or billion tons of plant-fixed carbon making its way into mycorrhizae per year is just a stunning number that I think it's fun to take on board and to think about, even if we don't try to augment that, how important that is to include in our models. So I will end with thanks to the many wonderful colleagues I get to work with and the funders who make it all happen. So thank you. Jess.
So, while I definitely like your idea that we should always be thinking about the active microbes,
yeah,
I am curious your thoughts on how we deal with that kind of
I completely agree that's important. I think I was describing to you earlier that we have a crazy Mediterranean climate in California where it's crazy hot and dry in August, and then 20 minutes later it starts raining, and then all hell breaks loose and the microbial communities change in 8 hours. And it's cold and wet in the winter, and anyway, just very different. And so we've sampled at all those different times of the year thinking, okay, if we take the active organisms at 9 different times of the year, we add them all together, do we get everybody? We haven't figured that out quite yet. I'm going to guess no, and not just because of relic DNA, because there's always some methanogens in there. There's always somebody who's just not happy, and we haven't quite captured their specific moment. And that's, I think, we all kind of learned about it as everything everywhere at one point, and that's why we do have such enormous diversity. We don't always sample it fully. But I would say why I'm still sticking to my guns on, I want to know who's active now if I'm trying to predict today's CO2 flux, today's nitrification rate, today's predation rate, CUE, whatever it is. Because yes, those other organisms tell me about the genomic and functional potential of the future, but they're not today's. I mean, I'll just say Mary Firestone, who I did my PhD with, used to talk about the piano keyboard as being the whole community. But the environment at any one point in time is only playing one chord, 2 hands, but 1 chord. So that's a limited scope of the rest of the environment. Yeah.
Okay, well, it's the end of the day, so let's thank Jennifer one more time. Now the moment we've all been waiting for. There's beer out there. Let me just, before you all head out, let me just say one more big thanks to all the speakers and all of the folks that made posters. And the last thing I will just say is that an unspoken theme throughout the day is that all of these talks, while many have been presented by more senior folks in the field, have clearly all been done by early career folks in these labs. And what I have seen today has been that the future is really bright, that the early career folks in this field are doing really, really wonderful work. So thanks to all the speakers for presenting all of this work, and thank you all for coming.